Welcome to the FDA Project-DataSphere Symposium on Rare Cancer Registries. I'm Bill Louv, President of Project-DataSphere, and it's my pleasure to be sharing the virtual stage with Dr. Julia Beaver. Julia is Chief of Medical Oncology at the FDA Oncology Center of Excellence, and it's been great to work with Julia and her colleagues to bring this symposium to you today. And Julia, I'd like now to invite you to share your thoughts about the importance of today's symposium.
Great, thank you. Good afternoon. I am very excited to be able to join in welcoming you all to this symposium. FDA and Project-DataSphere have a great history of collaboration with numerous past and ongoing projects working together towards advancing innovation in drug development. FDA and PDS have sponsored 8 past symposia focused on targeted research topics ranging from immune-mediated adverse events, artificial intelligence, big data, and immuno-oncology endpoints. This 9th symposium really hopes to address critical questions in the field of rare cancer registries with an ultimate goal to drive improvements in patient treatment, bringing safe and effective drugs to patients with rare malignancies in the most efficient and expeditious manner. This area is of particular relevance as it's clear over the last few years that improvements in molecular profiling and scientific knowledge have fostered a better understanding of biology and disease heterogeneity, resulting in rare tumor subtypes forming out of more common malignancies. And this has resulted in increasing levels of interest in rare cancer drug development, be it for cancers with rare mutations or rarer cancers that may not be as well understood with limited treatment options and high unmet medical needs. From a regulatory standpoint, it's critical to understand the natural history of disease for rare cancers, and we've been able to leverage registries to an extent in the past for regulatory purposes. However, with information technology breakthroughs and more innovative data science, cancer registries represent a real-world data source that can offer many potential benefits, such as the ability to collect and pool cancer data for individual patients, follow patients longitudinally for cancer-related outcomes and safety, and thereby achieve a better picture of the natural history of the rare cancer. Also allow for a more complete risk-benefit assessment of applications from a regulatory standpoint. Also, rare cancer registries could help with trial planning and estimation of effect, and knowledge gleaned from those registries can be very helpful with this regard to study design. Registries can also be employed for study recruitment into research and interventional studies, either for rare cancers or biomarker subgroups of disease, where patients would know their mutation, be notified, and then empowered to join appropriate clinical trials as they open. Registry data can also be linked with patient-reported outcome data and facilitate a more patient-centered approach. And ultimately, rare cancer registries can supplement and complement more traditional clinical trial data, as well as drug development programs, potentially for use as external data sources. Opportunities still exist to better standardize and harmonize data elements, ensure data are relevant, robust, and reliable, and improve startup of broader registry networks. And patient involvement in rare cancer registries can assist with this and in driving the field forward, and will be critical to expanding the regulatory uses for registries. We are thrilled to be able to welcome you all to the conversations today, which will cover a lot of ground, going into details on the impact of registries on clinical practice and clinical decision-making, as well as the ability for rare cancer registries to advance drugs in development and support regulatory review and ultimate drug approval. I want to thank all of the participants, FDA and Project-DataSphere, for supporting this critical discussion. And I will now turn the opening remarks back over to Dr. Bill Louv.
Thanks, Julia. You covered that extremely well. I'll try not to go over any of the same ground. That was really great. As I say, we're really proud of this collaboration and the 9 seminars that we've had starting in 2015. And we see ourselves as collaborators, conveners, and catalysts, bringing together scientists from academia, from government, and from industry to work on these opportunities together, to learn from one another, to have these discussions, and to identify actions where we can have positive impact on outcomes for cancer patients. And these symposia have really influenced the scope of research programs at Project DataSphere. So as Julia was talking about the topics of recent symposia, there are research programs at Project DataSphere for each of the last 4, or corresponding to each of the last 4 symposia. So external control arms, IRAE, applying AI to assessing tumor progression, and then of course today looking at the creation of registries for rare diseases. Here's an early advertisement for the next symposium, Symposium 10, tentatively scheduled for spring of 2021, and the topic will be data sharing. As Project DataSphere, in addition to its research programs, continues to operate an open science, open model data sharing platform. And that platform continues to grow with respect to patient level data that's available on the platform, number of registered users, and the number of peer-reviewed articles that have been published based on the platform, which is now past 85. So it's still a very busy activity. And the next symposium, we'll see, can we take this to yet another level of productivity and output? So we have a great agenda today, and it's now my honor to introduce today's keynote speaker. And that keynote speaker is Dr. David Fajgenbaum. And some of you will know some of David's story about how he went from a healthy athlete to a critically ill patient virtually overnight. And his illness stumped expert after expert who could not figure out how to treat his condition. Well, David, he's a force of nature, as you will get a taste of here during his keynote address. He sort of took control, and he became a leading expert on Castleman's disease, which is the disease he's afflicted with, and generated new sites regarding the disease and new insights about how to find effective treatments. And today, Dr. Fajgenbaum is an assistant professor of medicine at University of Pennsylvania. He's the associate director of the Orphan Disease Center. He's director of the Center for Cytokine Storm Treatment. He's the co-founder of the Castleman Disease Collaborative Network. And he's the leader of 18 clinical research studies, including a natural history registry of Castleman's disease. So I think it's clear that David is the ideal keynote speaker for our symposium today. David, over to you.
Bill, thank you so much for that introduction. What an honor it is for me to be a part of this really important meeting and to be able to share a bit with all of you about my journey. I'm going to begin by sharing my screen. So, Bill, my talk today, framed in the context of this really important meeting, is about cancer registries and personalized medicine. Personalized medicine is a term that we hear a lot in medicine. I'm going to be sharing my own take on personalized medicine. So today I'm going to share about really 5 perspectives that I think are critical when we think about taking down cancer and really generating the data to do that. The first is that of a caregiver. This is a picture of my mom and I shortly after her diagnosis with brain cancer. Watching her battle and watching the way her doctors took care of her inspired me to want to go into medicine shortly after she passed away from battling glioblastoma. A few years later, I was a third-year medical student at the University of Pennsylvania, hoping to become an oncologist and treat patients like my mom, when just as Bill said, I became this critically ill patient, seemingly out of nowhere, with multi-organ failure, and even had my last rites read to me when my doctors didn't think I would survive. But it was realizing that I didn't just need to survive, I needed to actually get involved in research if I wanted to live long term. And so I started searching for treatments that could maybe help me. And then I realized that it wasn't something I could do on my own. It was something that I would need to become an advocate, to connect with other physicians, researchers, and patients to really make progress. So I'll be sharing these 5 perspectives today as we think about rare cancer registries. This is just a schematic that I wanted to highlight how, in order to make progress, we really need the patient perspective, the physician perspective, advocates, we need researchers, and we need loved ones of patients, caregivers, to be a part of the conversation, to work with FDA and with pharmaceutical companies so that we can really make a difference for individual patients. And also that we can think beyond an individual patient towards the entire disease field. And what I'm a huge proponent of is actually learning from one disease field and applying it to others. So thinking about how do you apply this network across diseases and things that we learned for, say, Castleman disease to rare cancers. So today I'm going to begin by sharing my personal journey, share about the collaborative natural history registry that I run for Castleman disease, how it's helped us to lead to novel therapeutics for Castleman disease, share about some applications beyond, and then finally close with some lessons learned. So as I shared before, I was a healthy third-year medical student, hoping to one day become an oncologist at Penn. And then out of nowhere, I started experiencing multi-organ failure. My liver, my kidneys, my bone marrow, my heart, and my lungs all began to shut down. And I was hospitalized due to a cytokine storm. My immune system was literally attacking my vital organs, but we didn't know why. We had no idea what was causing it. Over the course of 11 weeks, I gained 70 pounds of fluid. I drifted in and out of consciousness. I even had a retinal hemorrhage that made me blind in my left eye. All of this without a single diagnosis. But thankfully, the diagnosis was eventually made within about 48 hours of having my last rites read to me because the doctors didn't think I would make it. And with the diagnosis of idiopathic multicentric Castleman disease, my doctors were able to begin treatment. IMCD is a rare immune hyperactivation disorder. We actually don't know whether it's caused by somatic mutation, where we would call it truly a cancer, a lymphoma, or if it should more appropriately be considered an autoimmune condition. It really sits at the intersection of autoimmunity and oncology. But because the immune hyperactivation is so deadly and we don't know how to stop it, I was given multi-agent chemotherapy, a combination of 7 different chemotherapies that completely obliterated my immune system, but actually started helping me to feel better. I felt better with every dose of Adriamycin, Cytoxan, etoposide because I was so sick from my immune system hyperactivation. And over the course of, this is almost 5 months into my disease course, you can see my dad and I sitting up. I'm just so thankful that the chemotherapy has kicked in and is saving my life. This is another picture of my dad and I a few days later. This actually happens to be New Year's Eve of 2010. And a few minutes after this picture was taken, we were walking around the hematology oncology unit, and we passed the family waiting room, and we noticed that there was a gentleman who was kind of swaying in his chair. He was clearly drunk on New Year's Eve. And on our next lap, my dad saw that this guy had fallen onto the ground, and so he helped him back into his chair. And he looked at my dad and I, and he said, "Thanks so much. Good luck to you and your wife." We were really confused at first, like, wife, what is he talking about? And then I looked at my belly and I realized that he thought that I was my dad's pregnant wife, which was a low point emotionally. But we laughed really, really hard. And maybe you can see how someone maybe thought that I was my dad's pregnant wife with the big belly from all the liver and kidney failure I was experiencing. About 7 weeks after that moment, I was finally discharged from the hospital. And you can see that I've still got a big belly from the liver and kidney failure. I lost all my hair at this stage. And this is a picture of me from just a few years before. I played college football at Georgetown. And I always say this is the worst before and after picture of all time. But if we could flip the order, it would be this amazing advertisement for Peloton or for Muscle Milk. But unfortunately, the pictures are in the wrong order. So about 6 months later, I was able to return to medical school. This time I was on an experimental drug, a drug called siltuximab, that blocks interleukin-6, one particular cytokine in the cytokine storm. And we were really hopeful it was going to keep me in remission. So when I returned to Penn, I got involved in the new orphan disease center that had been started at Penn. But unfortunately, about a year after getting out of the hospital, I had a full-blown relapse, this time on the only drug in development for my condition. And my doctor explained to me that there were no more drugs in development and that I had literally run out of options. And so I turned to my dad, my sisters, and my girlfriend, and I promised them I would dedicate the rest of my life, however long that may be, to trying to identify a treatment, maybe even a cure for my condition. So the first thing I did was find a lab on campus where I could get a little bit of space and start conducting laboratory research. I could get access to my own blood samples. I couldn't get access to too many other samples at the time, but thankfully a colleague provided me some space in her lab. And I started a foundation called the Castleman Disease Collaborative Network because I knew I couldn't make the progress we needed on my own. I knew that I needed to bring together physicians, researchers, and patients to advance research, and I knew that we needed to take a different approach. There were already 2 foundations in the Castleman space that were doing research the traditional way, raising money and then putting out requests for proposals. We wanted to change that, and we came up with a new approach we called the collaborative network approach, where physicians, researchers, and patients crowdsource and provide ideas towards research, and then we prioritize those ideas and then go out and recruit the best people in the world to do the work. And this has really just been a game changer for our community. But there's an important part to that message of prioritizing and then seeking the right people to do that. And that's that we couldn't just wait and hope for the right research to happen. And this is a quote from my book, From Chasing My Cure. Hope cannot be a passive concept. It's a choice and a force. Hoping for something takes more than casting out a wish to the universe and waiting for it to occur. Hope should inspire action. And when it does inspire action in medicine and science, that hope can become a reality beyond your wildest dreams. For me, this was the moment in my life where everything changed, where I went from hoping that some researcher somewhere would figure out a treatment that could save my life to saying, if I'm going to hope for it, I need to spend my life and do everything I can to try to find that treatment rather than hoping for someone else to do it. So when I got out of the hospital, I tried to get a sense for what's going on in the idiopathic multicentric Castleman disease space. And I found that there was about $10,000 a year being invested into IMCD research, which, as you all know, is too little to make any sort of meaningful progress. The NIH had never given any funding towards IMCD research. There were no patients in a registry because no registry existed. There were 2 advocacy organizations I mentioned, but there was no sort of research agenda or centralized research plan. There were no cell lines, animal models, biobanks. The disease, as you can imagine, was very poorly understood. There were no diagnostic criteria, no treatment guidelines, no blood tests you could run to predict who's going to get better on chemotherapy or who's going to get better on IL-6 blockade, no FDA-approved treatments. But thankfully, there was that one drug in development, siltuximab. The drug that I mentioned was tried on me under emergency compassionate use from the FDA, but unfortunately, it did not work for me. So a little bit about IMCD and kind of what we were up against. So I mentioned IMCD is a cytokine storm disorder where there's incredible immune hyperactivation, but we don't know why that happens. We don't know if there are autoimmune mechanisms that are leading to the immune hyperactivation. We don't know if there are neoplastic cells, and this really is a lymphoma, or maybe there's some pathogen that we just haven't discovered yet that's igniting the cytokine storm. But thankfully, we did learn over 20 years ago that interleukin-6 was critical to the cytokine storm because Kazuo Yoshizaki, who's photographed here, discovered that interleukin-6 was elevated in a number of IMCD patients in Japan. Based on that observation, he wanted to develop a drug, which many of you all will be familiar with, called tocilizumab, that blocks the receptor for IL-6 with the intent of treating idiopathic multicentric Castleman disease patients in Japan. And I'd heard a rumor that before he tested it on patients, that he tested it on himself. And so when I asked Kazuo, I said, Kazuo, I heard that you gave yourself tocilizumab to prove that it was safe. And he said, no, I didn't give it to myself. The nurse, she gave it to me. So Kazuo Yoshizaki made the discovery that IL-6 was a target. He developed the drug. He gave it to himself to prove that it was safe. And then he went on to give it to other humans in a phase 1 trial in Japan, which led to the approval of tocilizumab in Japan for idiopathic multicentric Castleman disease. And of course, you all will be familiar that tocilizumab, or Actemra, came to the United States and was eventually approved for rheumatoid arthritis and also for CAR-T therapy, the cytokine storm that occurs with it. That work inspired work by Janssen Pharmaceuticals to develop a drug called siltuximab, but rather than blocking the receptor for interleukin-6, it actually blocks the ligand directly. But we know from research that if you give a patient tocilizumab or siltuximab, that about 1/3 of patients will benefit from IL-6 blockade. Unfortunately, For the 2/3 of patients who don't benefit from IL-6 blockade, patients like myself, we don't know what else to do other than using cytotoxic chemotherapy. So my research focus really became, let's tease out what's happening biologically in the IL-6 refractory patients so we can identify drugs other than chemotherapy. Many of you all will know that this drug that Kazu developed was used to save the lives and has been used to save the lives of thousands of CART therapy patients who got cytokine storms after their CART therapy. This is a picture of Emily Whitehead, the very first pediatric patient to ever get CART therapy, who was dying in the ICU just next door to me at the Children's Hospital of Philadelphia before tocilizumab saved her life. This drug developed for Castleman disease many years ago, saving Emily's life and likely also preserving the CART therapy program. I suspect that if young Emily had passed away, that it really could have led to in addition to incredible heartache for her family, really slowed down the entire cellular immunotherapy program worldwide. Many of you all may be familiar with the use of tocilizumab off-label for COVID. The cytokine storm that we see in idiopathic multicentric Castleman disease is actually very similar to the cytokine storm that occurs in COVID-19. And as a result, many individuals have used tocilizumab off-label. And it actually got a lot of interest early on. And I think many of us are very hopeful that tocilizumab was going to be the solution for the most sick patients. And actually, a number of trials suggested that. But as time has gone on, and larger studies have been done, phase 3 studies, it's less clear whether it's helpful. There was a very clear negative result. And then more recently, just in the last couple of weeks, there was another result suggesting benefit. So the jury's still out. But as we shifted gears towards understanding what could we do to better understand treatments and therapeutic options for Castleman disease, I again knew that I couldn't do it alone. And so I was a medical student at the time. I enlisted a number of my classmates to join our team. And then I was a business school student and enlisted Wharton students to help me to build this team and to take on Castleman disease. In this picture, you see we're doing what we call the Castleman Flex. The disease is named after Benjamin Castleman, but we've kind of repurposed it as a warrior. And if we were all here, if we're all together today, I would have asked all of you to do a warrior flex, but I hope you'll do a warrior flex by your computer. I also knew that we need to engage the entire global physician and researcher community, and we've done that. And really, it's through connecting with these physicians, scientists, and patients all over the world that we've been able to lay out the right research agenda for progress. This is one way I like to think about drug development, particularly drug repurposing. It's around identification and validation of drug candidates and therapeutic targets. There are a number of ways to do that. It's about really harnessing and leveraging real-world data, whether it's off-label drug use, or well, it's always off-label, but intentional off-label drug use, or maybe observational studies where individuals are exposed to a drug that's not for the primary indication. Of course, moving forward to interventional studies, and then working towards FDA approval. Of course, if it's a not yet approved drug, it requires FDA approval. But if it's a drug that's already approved for something else, there are a number of conditions where we're doing really high-quality trials and it can really change clinical practice without actually having a label change. But there is one part of this that I feel is essential, and that's why I believe I was asked to speak today, and that's that it is so critical to utilize the data that exists to establish the natural history of disease and to track treatments that are used for that condition. We heard earlier about how natural history studies and registries can actually help to inform endpoints for clinical trials. That is absolutely critical. And in addition, what was also mentioned is that registries and natural history studies are often used as post-approval measures to actually phase 4 studies after approval to track the use of these drugs. And so I'll be sharing about our experience with Castleman disease that's actually meeting both of these objectives. So turning over towards our Castleman disease space. So a few years ago, we decided we wanted to launch a collaborative natural history registry for Castleman disease, and there were a few reasons for that. The first is that the EMA required Janssen Pharmaceuticals, which was the makers of that IL-6 blocker siltuximab, to launch a registry back in 2014. It was not required by the FDA, but EMA did require it. In parallel, about 2 years before, the CDC and Scientific Advisory Board prioritized launching a registry as a priority for the field. And so the CDC and UPenn and Janssen worked together to come up with this tripartite agreement where we would both meet the EMA requirement that they had imposed on Janssen while at the same time harnessing incredible amounts of data to better understand the natural history of Castleman disease and therapeutics. And so just kind of boiling it down, we wanted to do this because this is a rare disease with a small patient population, so limited data previously available. It was poorly understood, and there was a lot of off-label drug use that we wanted to get a sense for, in addition to meeting the CMA requirement. So the first thing we did was try to get a sense for, well, how do these registries typically collect data? What's the typical registry approach, data collection approach? You're likely all familiar with the kind of classic patient registry where patients go online, they enroll in high numbers because you can access patients quite easily online. Because it's patient reported and because the medical record is not available often, there's a small amount of data that you can definitively get from a patient through a traditional patient registry. You can't get every laboratory test ever run, every pathology report on a patient. You can get patient-reported outcomes that are critical, but you end up getting a smaller amount, a quantity of data than you would from the medical record, but it's low cost. The next approach is what we consider physician-directed, and this is the traditional natural history study, where you select 10 sites and those 10 doctors enroll all the patients they see with that given disease. So you have a low number of patients because if it's a rare disease, you'd need to have 100 sites or 1,000 sites to really enroll high numbers. So you're going to have a smaller patient enrollment, limited scope, but you're going to get increased quantity of data. The doctor, the physician is going to have access to laboratory data, imaging data. There's a lot more data that can be entered. And of course, the cost is higher. You have to pay the physicians and the sites for their time. The third approach involves manual EHR extraction, where you actually manually pull data from the electronic medical record. You're going to have low patient enrollment because you have to basically reinvent the wheel for every single EHR. You can do this at Penn, and then you can do it at Sloan Kettering, and then you can do it at each institution, on and on. But the numbers are going to be low because it's an institutional focused approach. The quantity of data is going to be huge. It's from the EHR, and the cost is expensive because it's a system by system approach. The fourth approach would be insurance claims data, leveraging ICD codes to find patients. And so now you're going to have a huge number of patients at your disposal. The quantity of data is kind of in between. There's, in some cases, you get a lot of data on use of medical resources. You don't necessarily get laboratory data and insights that'll come from the EHR, and it's pretty expensive. And so when we looked at all these options, we said, well, let's come up with a hybrid, what we call a natural history registry, where we combine the best of the patient-reported piece with the best of the physician-directed piece. And I'll go through that now. We call this approach, and we call it the Accelerate Natural History Registry. And we've been doing this for the last 4 years, and I'm really, really pleased to share the way that we do it. So it's a hybrid approach in that patients enroll online, they consent online. As soon as they enroll, they provide patient-reported outcomes, and they continue to provide PROs every 3 months. But once they enroll, they give us permission to access their complete medical records. So we reach out, we get the complete medical records sent to the University of Pennsylvania. At the same time, we also request any biospecimens, lymph node tissue, bone marrow, any samples that are left over or that are available. Then we have manual data extractors. We have 3 full-time employees who go line by line through every single medical record and extract out thousands of data points per patient. That data is then shared with an expert panel that reviews the diagnosis and also confirms the treatment responses. And then we use that curated information to glean clinical insights about Castleman disease and also to correlate this really extensive clinical data with each of those biospecimens. So now you don't just have a lymph node from a Castleman's patient, you have a lymph node from a Castleman's patient, and you know the entire clinical history that led up to that lymph node resection and also after that lymph node resection, what drugs were used, what worked, what didn't. It's an incredible amount of power that comes from that. And so kind of going back to that framework that I showed at the beginning of my talk, we're bringing together patients, loved ones, researchers, and also advocates through the Castleman Disease Collaborative Network, while at the same time pulling together and including the physician voice through our partnership in doing it at the University of Pennsylvania, have had a wonderful partner in Janssen Pharmaceuticals, and then also worked very closely with the European Medicines Association, really bringing together these 7 stakeholders to make really exciting progress. So what is some of the progress that we've made? Well, we've enrolled over 400 patients in our first 4 years. And for each of those patients I mentioned, we have extensive longitudinal data. So if we enroll a patient today, We get all of their historical medical records, enter those data in. We also start getting data prospectively that we enter in, so really extensive data. We get between 3,000 to 4,000 data points entered per patient, which takes days and days of medical extraction by these extractors. On average, there are about 683 clinical and imaging data elements per patient. And as I mentioned, there are about 3,000 plus other data elements that are collected on a per patient basis. And we've used this information to discover a brand new subtype of Castleman disease with really important treatment implications, a group of patients who were previously included in multicentric Castleman disease that really should be treated differently from multicentric Castleman disease. We've amazingly identified over 40 different drugs have been given off-label to Castleman's patients. There's only one drug, siltuximab, that's FDA approved, but over 40 other drugs have been used. And there's a long tail. There's a select number that have been used quite a bit, and then there's a number of drugs used just one or two times. But it's so important to collect this data. Previously, it wasn't being collected anywhere. We've used this data to enable a number of translational research studies. I've listed them on the right of the screen, but I'll just highlight a few of the things that these data and these samples have unlocked, and that's to look into the role that a pathogen, a novel pathogen may be playing, to look into germline genetic studies, somatic mutation studies, autoantibody work, pathway investigations, proteomic analyses, single-cell RNA sequencing on peripheral blood cells, morphometric analysis of lymph node tissue and bone marrow, and importantly, bringing all this together, this has led to the discovery of two novel therapeutic targets, really emphasizing the importance of having natural history data tied with biobank tissue and then using it. I get frustrated when I hear about people banking samples. The goal is not to bank samples. The goal is to collect samples, to use them for research so that we can help patients like myself. So let me highlight a couple of these discoveries. The first, and this is a picture of my girlfriend at the time, Caitlin, and I, around the time that I graduated from medical school, on my medical school graduation day, in fact. And then this is a picture of the two of us a few months later. In the time between these photos, we got engaged to get married, but unfortunately, I relapsed and nearly died for the fifth time battling idiopathic multicentric Castleman disease. So despite the work, despite setting up registries and setting up omics studies, that progress wasn't fast enough to help me. And as I said, I nearly died for the fifth time, but thankfully multi-agent chemotherapy saved my life. But I realized that if I wanted to make it to Caitlin and I's wedding date, which was May 24th, 2014, that I would need to do everything in my power to try to identify a drug that could maybe save my life. And I'll go through this slide quickly, but just to highlight the top, these are the drugs that have been used in my case. And this is data from multiple different hospitals. I was hospitalized in a number of different places, different healthcare systems. So pulling all this data together into the registry meant that we could harmonize information on laboratory values, cytokine panels from multiple institutions. So I'm showing you data from the registry, highlighting the drugs that were used at the top, and also you can see the really intense ebbs and flows and ups and downs of key laboratory values. But the point is, a number of drugs were tried, but it wasn't until I got multi-agent chemotherapy with DDT-ACE that I had a nice year-long remission and then another year-long remission. But here I was, fifth flare of this disease, really without options. And so what do you do when you've run out of therapeutic options? Well, in my case, I turned to the data. I turned to the data in this system to understand the longitudinal data from these multiple institutions to identify these trends. And the first thing we found was that we've been running cytokine panels every month on my samples, and we found that VEGF, or vascular endothelial growth factor, which is a marker of angiogenesis, and also soluble IL-2 receptor, which is a marker of T-cell activation, were both very elevated in the weeks leading up to my relapse. The gray bar here is the relapse, and you can see these cytokines increasing in the weeks and months leading up to the relapse. So we thought that maybe this indicated that VEGF and soluble IL-2 receptor, or T-cell activation, could be important to Castleman disease pathogenesis in my case. Now, again, if we weren't bringing together data from multiple institutions, multiple different sources, we wouldn't have picked up on this trend. Thankfully, I had stored some serum and also blood cells, so PBMCs on myself from my fifth relapse. And so I went into the lab and I began performing experiments on those samples. And I was able to confirm that, in fact, VEGF and soluble IL-2 receptor were two of the most elevated proteins when we measured 1,300 different proteins. And I was able to confirm through flow cytometry that there was a tremendous amount of T-cell activation in those samples. Importantly, the VEGF story around vascularization certainly was consistent with my clinical picture. So I had increased vascularity in my lymph node tissue. I had these cherry hemangiomas on my skin, which are blood moles that were popping up, likely related to elevated VEGF. And I had this tremendous amount of fluid accumulation, which can also be secondary to VEGF. So we wondered, is there something that's involved in all three of these phenomena that we've observed? And so we ran the proteomic data through a number of pathway analyses tools. And what we found was the mTOR pathway came up a number of times. mTOR is a really important pathway involved in cellular proliferation and communication within the immune system. So it kind of fit the bill, but we wanted to confirm, was the mTOR pathway truly elevated and was there truly increased activation? And so I ran an experiment on a number of normal lymph node samples. And so if the cells have increased mTOR activation, you're going to see that they're brown. And if they don't, they're going to be blue, counterstained. And you can see this is what a normal lymph node looks like with a normal amount of mTOR activation. And then this is my lymph node, and you're just really blown away by the amount of mTOR activation in my tissue. So we didn't know that blocking mTOR would be effective. There was no guarantee that if we blocked mTOR it would be effective. But based on these data and based on all of the work leading up to finding mTOR, we thought that if we could block the mTOR pathway with a drug like sirolimus that's been around for 30 plus years, that maybe we could control this condition. And so we began trying sirolimus on me 81.06 months ago that I've now been in remission. And I say 81.06 months because I know that I can't round up. I don't know if I'm going to make it to 82 months in remission on this treatment, but I also refuse to round down. It hasn't been 81 months. It's been 81.06 months because I'm just so thankful for every moment of remission that I've had and so grateful for all the people who helped to make this possible. The New York Times called this "Doctor Cure Thyself," which I think is a bit of an overstatement. I think it should be doctor helping himself a little bit right now and hopefully for a lot longer. But really importantly, it did enable Caitlin and I to get married. And this is a picture of us on our wedding day, and you can see my hair grew back just in time for this picture. And then a couple years ago, we had our first child, Amelia, photographed here. And this is her just a few weeks ago on her second birthday. We've gone on to show that the mTOR pathway is important in other patients, showing that it was increased and that mTOR inhibition was effective in 3 patients, going on to show that it was increased in 26 patients earlier this year, and identifying a potential candidate driver for why the mTOR pathway is increased. And this has led us to give this treatment to other patients. This picture of Joey, who was really struggling and benefited very well from sirolimus, whereas unfortunately Lisa and Sergio, who didn't respond to first, second, or third line therapy, did not benefit from sirolimus, and they push us and drive us and inspire us to continue to work harder. So this is where we were in 2012, and I'm going to superimpose where we are today. We've now raised about $1.5 million, which led to an additional $10 million in external funding. My lab now has the first NIH grant to study Castleman's. We've got over 1,200 patients in our registry. We went from there being 2 foundations to 3 at one point when we started, and now we have just one unified organization. We've won biobank. We're answering questions about the disease. We finally published diagnostic criteria and treatment guidelines, predictive biomarkers. We now have that one FDA-approved drug, siltuximab, and now there's 2 more drugs in development. The drug I'm on, sirolimus, which is undergoing a clinical trial right now, and then JAK inhibitors, which are also undergoing development as well. So in the last couple minutes, I did want to highlight that this idea of uniting to advance progress is something that has happened across medicine and across oncology in particular. The Cordoma Foundation is an example of a great group that's brought together physicians and researchers for scientific discourse. The Rare is One project that Chan Zuckerberg Initiative is leading is intended to collect and crowdsource research ideas, just like we've done for the CDCN. Count Me In is a great example of working directly with patients to get samples and medical data. The TAPER study from ASCO is a great example of systematic tracking off-label treatments. And then when COVID started, my team and I at the Center for Cytokine Storm Treatment and Laboratory launched what we call the Corona Project to systematically track treatments used against COVID-19. You can check out the link at the bottom of this page to see the database, but our goal and what we have is the world's largest database of all treatments used for COVID-19, many of which are Castleman's treatments. But amazingly, we hear about the same 5 drugs in the news every day, but there have actually been over 300 different treatments given to COVID-19 patients, and anyone and everyone can access this. We're sharing the data directly with FDA and the Cure ID app in particular, and we'll be thrilled for anyone to check it out. I don't have time to go through this in depth, but I can tell you that we've made some really important observations related to the immune response, that it's not just about being too strong of a cytokine storm, but it's about having that modulated response where you're looking for it to be right down the middle. This image is, and this is me just closing with lessons learned, is trying to show that there are about 10,000 human diseases. And unfortunately, we have treatments that are FDA approved for only about 1/4 of them. So 1/4 of human diseases have an approved compound, the other 3/4 do not. And so what I really am a big proponent of is thinking about all the drugs that are already FDA approved, like sirolimus, like ruxolitinib, like these drugs that we've identified as being candidates from our research. How many of these old drugs can be used in new ways? This is a cartoon of an old dog reading a book about new tricks. And I think we need to think about that and all these old drugs that can be used in new ways. And how can we, patients, physicians, researchers, advocates, FDA, pharmaceutical companies, how can we work together to help individual patients, to help advance for particular diseases and help to advance across diseases? And I really challenge and encourage all of you to think about ways we can bring those stakeholders together. This is my last slide, just closing with some final thoughts. The first is that I hope that my experience and my talk has shown the incredible power that comes from bringing patients to the table and actually letting patients be a part of driving forward research, crowdsourcing research, pushing forward the science, and how important this is beyond Castleman disease. The second is that hope can have many different forms. My life fundamentally changed when I went from being a hopeful person to being someone who turns my hope into action. The third is that solutions can be hiding in plain sight. The drug that I'm on was at my nearby CVS for all of those years that I was in and out of the ICU. How many other drugs are sitting at our neighborhood CVS while many of us battle deadly conditions? And I think that it should be something that we all work towards, figuring out how many of those drugs are already out there. The fourth is that we need to systematically capture data. If we hadn't been collecting data from across institutions, I would have never picked up on the trends that we saw within cytokine levels, also clinically within my case, that helped to lead us towards sirolimus. I'm sitting here today because of a drug that was already FDA approved, because of data that were already out there. They just had to be harnessed to gain these insights. And the last comment is that it really takes an army. My book's called Chasing My Cure, but I think we should have called it Chasing Our Cures, because it hasn't been me working on my own. It's really been an incredible team. And I hope all of you watching today will become a part of this team. We'll advocate for drug repurposing. We'll advocate for involving patients and the entire community in research and help to spread the word about Chasing My Cure. I saw PDS is giving out copies of Chasing My Cure. I hope you'll read through it and help to spread the word about this journey. I wrote Chasing My Cure because I learned so much about life and living and hope and resilience from nearly dying, and it really means a lot to me to be able to share that message of hope with others. And as I shared, it has not been an effort that I've done on my own, and I do need to thank my team, many of the partners that we've worked with, and also funders for making this work possible. Thank you guys all so much for the opportunity to share my story.
David, thank you. It's such an inspiring story, and it's so motivating to see how you used hope to inspire tangible actions and real progress. It's amazing. We're all in awe of this journey, and thank you for sharing it with us. We really appreciate it.
Thanks so much, Bill, and thanks for helping to turn so many other people's hope into action through the work you guys do. It really, really means a lot.
Thank you. We're now going to have a short pause before we go to panel 1, but please don't run off. Stay by your computer. It'll be a short break, and during that break, there'll be important information on panel 1, including how you can pose questions to the experts.
Well, welcome back, everyone, and thank you for joining our first panel discussion. First of all, I would like to really thank as well Dr. Feigenbaum for a remarkable and inspirational story that he shared with us. That certainly inspired me and I think raised a number of the issues that we'll discuss in the remainder of today's sessions, including the two panel discussions. In this first panel, we'll explore rare cancer registries and their utility in clinical decision making. I'm David Reese, the head of R&D at Amgen. I'll be the moderator of the panel, and we're delighted to have join us today Drs. David Miller, Gregory Riemann, and Ting Yu, who will offer perspectives from academia, the FDA, and industry, respectively. Before we dive into the discussion, I'd like to start with having each panel member provide a brief introduction, and perhaps, David, we can start with you.
Good afternoon. I'm David Miller. I'm a medical oncologist and dermatologist at Massachusetts General Hospital. My clinical and research focus is in cutaneous oncology. I am the director of the Center for Merkel Cell Carcinoma at MGH and co-director of our multidisciplinary non-melanoma skin cancer clinics. I've been working with PDS for the last 2 years on developing a registry for Merkel cell carcinoma. I co-chair that task force with Mike Wong, who you'll meet in panel 2, and it's a real pleasure to be participating in this along with Dave, Ting, and Greg.
Great, thank you. Greg? Yeah, you appear to be muted, Greg, so why don't you go ahead and unmute? There you go.
Hi, I'm Greg Riemann, pediatric oncologist. Sorry for the technical snafu there. Currently Associate Director for Pediatric Oncology at the FDA's Oncology Center of Excellence. I have a long history before coming to the FDA in clinical investigation in childhood cancer with a special interest in new therapies for children with acute leukemia. And also in drug development for childhood cancer. So it's great to be here. Had wonderful experience over the years working with Project DataSphere. Happy to be part of this.
Great, thank you. And Ting?
Hi, my name is Ting Yu, by training Hemong guy. Currently working for EMD Strano. I am the indication lead for Merkel cell carcinoma and the GTN. Both are well qualified rare disease, I believe. So glad to be
here. We're really looking forward, I think, to a rich discussion. As we mentioned, Dr. Feigenbaum introduced and touched on a number of concepts, but I thought perhaps we could start by going back to basics and simply start with a simple question. What is a rare cancer registry? Is it simply a database? Is it more than that? Are there flavors? To give all of our participants, a sense of the span of what we mean by the term registry. So David, perhaps we can start with you and get your perspectives on this question.
Sure, happy to. And I will start with basics as well. I've always liked the adage that if someone has said it better, then use their quote. So I'll quote E.M. Brooks, 1974, the current and future use of registries and health information services to sort of lay the foundation. He defined registries as a file of documents containing uniform information about individual persons, collected in a systematic and comprehensive way in order to serve a predetermined purpose. So a cancer registry, as I see, is an information system designed for the collection, storage, and management of data on persons with cancer. And I think about it in a couple of different flavors, to use that term. You can have population-based registries, hospital-based registries, and then special cancer registries, which I've been a part of, where the goal is to collect and maintain data on a particular type of cancer. And to also, I think, address your last question about the difference of a registry and a data set, I would say, and interpretations, of course, vary, To me, the difference is a registry is a special type of data set that has a longitudinal component that is critical for the study of the natural history of an illness. So it is that ongoing nature, the continued curation over time that kind of gives a registry its distinction from just a plain data set that may be static over time.
Great, thank you. No, that's fantastic. Ting, maybe from the industry perspective, your view of what is a registry. I know you've had experience in this regard.
Yep. Thank you for the opportunity. Quite frankly, for the industry, used to be, or traditionally, it provides safety signals for the drug, especially as Dr. Fajgenbaum actually referred to the phase 4 studies and used to be used for monitoring AE events and to validate the efficacy and the safety information, mostly focusing on safety. However, in the current practice, especially in oncology, that's how it can be used, how it can be informative data set for everybody, for prescribers, for industry, and for scientists in the lab. It's actually a treasure trove. That's what I can see. David had already touched upon the longitudinal aspect of it, and that actually will be very informative in terms of a sequencing of the treatment, concurrent treatment versus sequencing. What will be providing the best efficacy? What will be providing the best safety profile? That's actually an excellent question to ask in the registry. And also I want to stress the point of comprehensive and systematic. As the keynote speaker already touched upon, and also David touched upon, sometimes those kind of information can be really ignite a fire by a sparkle of a biosample study and a cytokine study. So this is actually could be a very informative.
Well, thank you so much. And Gregory, from a regulatory perspective, any thoughts on sort of a basic definition of a registry?
Basic, I think David and Ting actually both contributed to a basic definition, if you will. I think one of the really critical issues in defining the basic necessities of a registry are sort of defining a purpose for the registry, the longitudinal nature of the registry, and most importantly, when aggregating information and particularly patient experience, doing it in such a way that that information is going to be operable, interoperable, and publicly available and utilizable. Utilizable by patients and families, utilizable by medical practitioners and caretakers, and ultimately, I think utilizable as we think about what is clinical benefit, and looking at the heterogeneity which spans rare diseases, except maybe those rare diseases which are due to a single gene defect, but given the variability in symptoms and clinical manifestations, I think having a registry and looking at potential interventions or just looking at natural history without interventions can help define what patients might experience and what patients might consider benefit. And that consideration really needs to drive drug developers and ultimately regulators.
Great, thank you. Well, you brought up sort of purposes and goals of a registry, which really, leads naturally to the question of what is the intent of a registry, or of course there can be perhaps many intents, from charting the natural history of the disease, as we heard from Dr. Feigenbaum as a first step, all the way through potential roles in regulatory outcomes. It'd be good to hear from our panelists again about their views on intents and how do you think about structuring a registry when you're thinking about the end in mind. So David, you've got some experience here. I think it would be valuable for you to share that with our audience because you've worked through a number of these problems yourself.
Yeah, thank you. It's a great question. And thinking back to 2 years ago when our task force first charted out our course, a key question, like the key question, was what are the objectives and applications? And over time that has evolved, not surprisingly. Initially, the idea was we had a rare disease in front of us that we all treated, but there was little data known about it. So understanding the basic characteristics of people who developed Merkel cell, and then understanding how the disease, how it progresses, what I would classify as understanding the natural history of the disease was initially, I think, the primary focus. And then, as we'll probably talk about later, that's evolved a little bit to also incorporate information potentially on best practices. I think Ting touched upon some of the best practice questions that we have. How do we sequence the available therapies that we now have through innovation at the sponsorship level and innovation at the regulatory level, to potentially getting information that could affect regulatory decision making, potentially affecting label modification through our data. And having those objectives thought out ahead of time is critical when you're developing your instruments in data fields in order to collect those important data elements to achieve either natural history understanding, best practice understanding, or potentially to influence regulatory decision making.
Great. No, thanks. And I want to return to that topic of best practices in just a minute. But Ting, interested to get your take here on intent, based on your extensive experience as well.
Sure. What I would think is actually natural course of a disease will change with different treatment options. A good example is actually Merkel cell carcinoma as well. So with the advent of avelumab and a checkpoint inhibitor in that particular rare disease, the natural course of the disease has been changed dramatically. So we raised the bar of the survival of the Merkel cell carcinoma, let's put it that way, and how high the bar is. And what will be the follow-up treatment should be improved upon, we don't know. So that's why even if we just a simple intent of capture the natural history of the disease is actually very informative for follow-on improvement and drug discovery as well. And let alone the combination, as I've already mentioned in an earlier conversation. So this is actually could be very constructive going forward. And for rare disease, actually the real world evidence could serve as a comparator arm. Quite frankly, because it will be very difficult to actually have a controlled study design for future regulatory approval. So this is just the couple points I want to
David
cover.
brought up a potential regulatory role of registries, and so we're interested in your perspective from the FDA. What do you view the potential role? Are there different roles? And I think this is a question that our audience will be intensely interested in. It's obviously a wide-ranging question, but we'd like to get your point of view.
Sure. Well, I think this topic will probably be also covered in the next panel. But I think as a start, I think there are multiple different opportunities and potential contributions that registries can make to the regulatory setting. Defining potential endpoints that might be used in subsequent clinical trials based on outcomes of patients and based on the natural history of the disease in patients. So just determining what is a patient's perspective on what would be called a clinical benefit or classified as a clinical benefit. I think from natural history would certainly give us an opportunity to look at endpoint development. One other thing I would go back to Dr. Feigenbaum's presentation about the linkage of registries with clinical data to biobanks. I think here again, there's a very rich opportunity to look at material that is available and usable again, and usable by scientists in multiple settings, sites, to actually look at correlations of different characteristics and natural history and potential biomarker development. As he very eloquently portrayed, not all patients with multisystem Castleman's respond to IL-6 inhibition. So looking for specific biomarkers that may predict a response to a new therapy, I think is an important outcome from
Great, thank you. And again, a lot of rich material, some of which our second panel will also explore. So we've talked a lot in theory so far, and perhaps we can move to actually some practical examples. If our panelists can give us examples of registries that they think are high quality and have utility. And then from there, perhaps we'll move on to what we can extract from best practices from those examples. So, let me start again with David here in terms of, when you survey the landscape from 1974 on, what sort of registries jump out at you, whether they're within oncology or outside of oncology for that matter.
Perfect. So, there are a plethora of examples where registry data has influenced my practice, and that's in part because I take care of very rare diseases, and Merkel cell, as has been said, is a great example of that. There are very few clinical research studies in general, period, in Merkel, but some of the ones that have influenced our practice come from, for example, SEER, the Surveillance and Epidemiology and Results Registry, which has been conducted through the National Cancer Institute. The National Cancer Database, or NCDB, is another rich source where numerous papers have been published using that data to influence our practice. And just for example, the current staging system that we use in Merkel cell, the AJCC 8th edition, which informs how we deliver prognostic information to our patients and how we ultimately adjudicate treatment decisions, came from a data set from the National Cancer Database. 9,000 patients with Merkel cell were used to construct the most recent staging system. So that's a good example of very practical applications. I have two more that I can briefly cite as well. So in Merkel cell cancer, there's a standard of care now to treat patients after you've had surgical excision with adjuvant radiotherapy. For most cases, there are some nuances to this, but that current standard of care of adjuvant radiotherapy in patients with MCC in part was supported from a 2007 publication of 1,100 patients from SEER, and then a 2019 publication of another 4,800 patients from the NCDB. My current practice of not using radiation for low-risk Merkel cell carcinoma patients came from a data set from the University of Washington, their single institution data set. influenced, really changed my practice. And then the final one of using baseline imaging in patients recently came from a publication that we actually just reviewed yesterday in our multidisciplinary journal club that again came from a single institution registry from the University of Washington, 500 patients, where they said, you know what, unlike melanoma, which is where we've been getting a lot of our practices, sort of inferring practice management of patients from melanoma into Merkel cell. Now, in contrast to melanoma, which typically, unless patients have regional and clinically regional involvement, we don't do baseline imaging. In Merkel cell, there's been heterogeneity in practices. And a recent publication that hasn't even been in print yet has sort of affirmed my practice of actually using baseline imaging in all patients with Merkel cell, because 1 in 3 patients that walk in the door with Merkel cell have either occult regional or occult distant disease. And that came from a single institution registry. So for rare diseases, registry data is pivotal in how we practice.
Great, thank you. Well, maybe we can go to Gregory next on this topic. You know, from your vantage point, you've seen a large number of registries. I wonder if you could share with us what you think some of the highlights are in terms of best practices.
I think as far as best practices, and one registry that particularly comes to mind, as a pediatric oncologist, all pediatric cancer is rare, but there are the rarest of the rare pediatric cancers for which clinical trials haven't existed, don't yet exist, but there are clearly opportunities. And one such registry is in a rare pediatric tumor, pleuropulmonary blastoma, which has, I think now, in over 20 years, basically accumulated a large natural history data on a sizable number of patients. There are only a handful of such patients diagnosed yearly, but it has really resulted in the development of a standard of care, if you will, from the standpoint of diagnosis, both clinical diagnosis as well as pathology, and now the banking of specimens to look for specific biomarkers that I think will actually result in the development of targeted therapies for this disease. So I think having a recognition that, and I think critical to best practice, is really a willingness to collaborate, participate, and understand the strength of coordination, cooperation, and collaboration. And in rare diseases, obviously that's critical. In childhood cancer, that's what's really driven the field over the last 5 or 6 decades. I think a realization and acceptance of that is
great. Thanks. Ting, anything you would like to add in terms of best practices based on your experience?
Sure. Before I joined the industry, I actually was working for Janssen on a CLL program, so I was involved in some CLL registry. What actually has been noticeable in terms of a registry in CLL is actually the contribution from the community practitioners. So we talked a lot about biobanking, we talked a lot about research, but the simple community, not cytogenetic, not biomarker-driven kind of a clinical practice need to be captured as well, because that represents some probably underserved patient population as well as part of the real world. So this is actually just the one point I want to just make sure.
Great, thanks. And maybe we'll start with you, Ting, on this next topic as well. We've got a number of questions coming in, and one of them I think we want to address because it will be of great interest is what are the challenges in incorporating data-rich data-rich elements in the registries? I'm thinking of omics data or immunophenotyping or imaging, which of course requires large amounts of storage and ways to handle the data and to ensure data quality. So Ting, perhaps you can give us your perspective, again, having established some of these registries and trying to encompass molecular and imaging data as well as basic patient data.
Sure. One thing I can think of regarding imaging is actually standardization. Several co-op groups actually had went great lengths to actually standardize all the imaging technique. That's actually quite crucial for the uniform readout for the results and assess the efficacy. So kudos to all the radiologists who is actually doing that. That's actually crucial for us as well. So that's one thing. Another part is actually personalized medicine. Personalized medicine really is actually individually tailored treatment. It's very difficult to harmonize, and hopefully the big data, we are talking about the PDS here. So the algorithm behind the big data readout could help for everybody to sift out some clue in the treatment. It's actually very heterogeneous in terms of cancer, in terms of response to drug treatment, and hopefully some clever algorithm can find
thanks.
us.
David, how are you thinking about data beyond basic natural history data, omics, imaging, other types of intensive data?
Yeah, so I think there is what I've been thinking about a lot lately is there's two opposing vectors when you're doing database or registry design. And there's the vector of data-rich data collection, and then the vector of feasibility. And a lot of times those are in opposite directions. I think all of us want to capture as much data as possible so we can move the field forward. And our task force has had lots of conversations and curation, rounds of curation in order to enhance the feasibility of this. So, we've taken the approach where we've had to early on prioritize which large data that we are able to capture first. So we've taken the approach of a multi-phased sort of data collection and release process. So initially, we thought the highest yield data the data element that found the right mix of feasibility and clinical utility, sort of capturing genomic data. So what we've done is develop an instrument in order to capture commercial and institutional NGS data. And then this is data that we can abstract from either a health record or from a report from a commercial vendor. It's most of the time it's unstructured data, sort of turn that unstructured data into structured data and then import it into our data collection tool. And that requires, some data science skills, it requires some resources, and also requires some skill. So we're hoping that our experience through capturing genomic data is going to inform our ability to capture the next phase of large-scale data, which, imaging is something that we're very interested in capturing serial imaging on patients in order to be able to perhaps inform, regulatory decisions based on imaging. Now, at this point, our imaging capture system is based on data entry of discrete fields. And for radiology reports, that requires a pretty intense resource in order to go through radiology reports and sort of do manual abstraction of those reports. With time and data science solutions, we hope to be able to innovate and be able to come up with text mining solutions in order to facilitate that. But at this point, we're still doing some manual abstraction. So in order to continue to make progress, we've sort of decided this would be a multi-phased approach where we prioritize capture of genomic information. And then once we do that, we'll sort of move towards the next phase, which would be imaging data.
Great, thanks. Well, Gregory, assuming we surmount all of these various challenges, is the FDA ready to, ingest large amounts of registry data? And it'd be very interesting to get your perspective on, how, we as a field can best utilize these data, but also, just from a regulatory perspective, how you'll actually handle it.
Good question, for which I don't have all of the answers. Is the FDA ready? I think the FDA has to be ready to at least work with industry and work with investigators and patient groups because we recognize the importance of these data and their potential contribution to future regulatory decision making. As some of the challenges have already been pointed out with respect to infrastructure and resource capabilities, the agency obviously has similar constraints. But I think working together as a field, we recognize that in order to get drugs to the patients who need them the fastest, we do have to look at new ways of demonstrating clinical benefit, new ways of determining short-term, long-term toxicities, and the old ways that we have done that in the setting of rare cancers really needs new think. And I think moving forward and working together, utilizing registry data is something that will become part of what we...
Great. No, thank you. Well, another question that's come in, which I think will also be of great interest to our audience, is the notion of open access. Project DataSphere, of course, pursues an open access model, but not all databases are open access, nor are all registries. Perhaps, David, we can start with you. The specific question is, how do we overcome institutional or organizational barriers, to submission to open access registries? It'd be very interesting to get your perspective from academia. Then I'll turn to Ting, who will give us perspective from industry, where, of course, other considerations also apply.
So it's a critical question, and one that our task force has been very thoughtful about. And there's no perfect solution to overcome some of these barriers. One methodology that we are moving forward with is sort of understanding that we all have various and different incentives. We've developed a multi-tiered data release platform that we are moving forward with. It's basically 3 tiers. So the first tier is a closed data collection and data access phase. So with our capture system, each institution that's led by a principal investigator is sort of the governor of that data set. And that individual institution has the ability to collect their own institution's data and then publish it as a single institution data set if they wanted to. Now, we realize that there are limitations to single institution data sets, and all the investigators in the task force realize that there are limitations to single institution data sets. So we know that most people are going to be motivated and incentivized to move on to the second phase, which is more of a closed access to the individuals who have aggregated that data. So for example, in the second phase, data that is captured by each institution is migrated over to the PDS platform. There's a period of time where that data is available to the institutions that have contributed that data. So there's a governing committee that would adjudicate the hypotheses that would be tested on that aggregated data. And then after that phase, we would open it to registered users on PDS. And we think that multi-step model provides the incentives that are necessary for investigators to participate. All of the investigators at each site have lots of different diverse teams that have different deliverables that they want, and having the ability to publish a single institution data set so that a graduate student can get a PhD dissertation, is an important option for people to be able to have, for example. And then having a data set that is aggregated gives incentives to the investigators. But then, of course, ultimately, what we want to be able to do through PDS is provide that information to a wider audience so that they can ask and test hypotheses that we haven't thought of, we won't think of, in order to move our field forward. So that's one of the methods that we're using in order to try to align incentives within the realities of academic medicine.
Yeah, no, that's great, and that's it's a very interesting thought around a period of restricted access for those who have contributed data and sweat equity, then followed by open access, a way to sort of compromise and provide appropriate incentives. Well, Ting, from your perspective, what are some of the barriers in industry? Of course, there may be proprietary considerations and other issues. How do you resolve those sorts of questions within an organization?
Yep. Thank you for the question. This is actually a challenge for every one of us. I cannot commit to say that we will disclose proprietary information that will be still protected. But frankly, by principle, it's actually to improve the health, improve the clinical outcome for the patient. So by that particular principle, we are committed to as much open as we could, especially on the safety signal side, on the efficacy outcome side. For sure, you have our commitment. Our senior management team is also team member, are also on this particular call. And I think he will agree to that as well, that it's a serious commitment from a industry partner that we will be open for the community, not only for the treaters, to the researchers, but also to the patient
really
in terms
perspective. We've got another question here that I think will be of great interest, which are, what are key considerations when you're using registries like SEER, NCDB to derive insights for broad populations? In other words, can we rely on registries for generalizability? What are our thoughts about that? Are there pitfalls? Are there ways that we can protect against false positive findings? Obviously, the larger the data set and the more clever the statistician, the more likely one will find associations. The question is whether they are true or not. And that's what interests all of us. And so I'm interested in this, sort of notion of how do we rely on registries for generalizability and the soundness of our conclusions. David, maybe we'll start with you here, and then we'll go to Greg, from an FDA perspective.
Yeah, I think those are great considerations, great questions. And, it's a multifaceted answer, of course. There are limitations to every single data set, and appreciating those limitations, whether they're missing data, biases, confounding, are critical. So having those registries be very transparent about how they have handled missing data, for example, or how they've handled bias is critical. And I think as a practitioner, one of the things, so at the core of clinical research is trying to minimize medical decision-making under uncertainty, right? On a quotidian basis, I am, and all clinicians are carrying out medical decision-making under uncertainty. And therefore, in order to minimize that, we need reliable inferences from data sets. And I think what at the core is, there is no single reliable data set, right? We need to pool multiple data sets and be able to synthesize and integrate inferences from each data set in order to ultimately take care of our patients, in order to ultimately make medical decisions. So there is a real need to have numerous registries in order to have, say, validation of the findings of one registry. So if we can support some of the findings in SEER or NCDB, with our registry, then that provides confidence in those inferences. And as you said, as we sort of grow our data sets, we get closer and closer to big N. We have more reliability on those inferences. So I think, to summarize, being very transparent about missing data, biases, and then also understanding that we need multiple various sources of those data in order to have reliable inferences to make medical decisions.
Oh, great, thanks. Gregory, from your perspective, what do you look for in terms of comfort with generalizability or in assessing the soundness of findings that may emanate from a registry?
Well, I think from the perspective of generalizability, and particularly since we are discussing rare cancer registries, I think a registry that draws from more than a single institution is clearly a superior registry, only because of or not only, but in large part because of the concern of unintentional bias that may exist, and especially when data elements may be objectively determined data coupled with subjective assessments. I think unless the registry is very, very well structured and that structure is made transparently known to potential users, then I think having a registry that draws from multiple centers, multiple treatment sites, patients from different parts of or geographically dispersed and represented, I think would give the agency more comfort.
Great, thank you. Well, another question that's come in relates to patient-reported outcomes, and I think we'll have time to address this final question. And it's appropriate that we return the focus to the patient, of course, because we want to improve patient care and patient outcomes, when we are constructing and utilizing registries. And one of the key things for patients, of course, are what we call patient-reported outcomes, that very broad category of data and assessments that include quality of life and many other measures. So the question is, how do we think about patient-reported outcomes as we're generating registries, and how do we use PROs to really inform us about the natural history of the disease, but how patients are actually really deriving benefit, for example, from potential interventions. So, David, why don't we start with you here, then we'll turn to Ting, and we'll wrap things up with Gregory. David?
So it's a great question. I think patient-reported outcomes are a key metric for healthcare delivery. I think that the question highlights a couple of things. One is that, when you're constructing a registry development team, it's really important to have a diverse group of experts on your panel. And you need to have domain expertise, of course. In the case of Merkel cell, you need to have dermatologic expertise, pathologic expertise, medical surgery, radiation oncology. But beyond that, you need to have expertise in data sciences, biostatisticians, epidemiology. And I think patient-reported outcomes is a good example of a niche that requires a lot of expertise. And, patient-reported outcomes are facilitated now due to some of the technological advancements in data capture systems. So the data capture system that we use, REDCap, has the ability to have surveys that go out to respondents or patients, and then they're able to fill out that data, and then you can insert that back in a structured form into your data set. So that is something that we haven't integrated yet. It is a plan for 2021 is to sort of figure out ways to optimize PRO assessments into our registry. But at this time, I haven't had the time to really dive into it. So a real nuanced discussion of PROs is a little bit beyond my expertise at this point. So I'd be happy to get some insight from the other panelists.
That's great. Thank you. Ting?
PRO, patient-reported outcome and the quality of life assessment is inherently part of the registry if industry is designing it for two reasons. One is actually just as our chair, David Rees, just mentioned, it's actually all about improving the quality of care. Another aspect is the payer. How are we going to justify certain cost of the healthcare to the payer system? That's another endpoint. And increasingly, the quality of life becomes a regulatory endpoint as well. I believe Greg can touch upon this. At least in Europe, this is becoming more and more
Thank you.
important.
Gregory, why don't you wrap things up from an FDA perspective on PROs?
Well, I think PROs are clearly something that are of great interest to the agency, and recognizing, appreciating, understanding, utilizing, hearing the patient's voice in regulatory decision making is really critically important. I think there are difficulties with validation of specific instruments that might be used. And I think another challenge is sort of global quality of life measures, which may be impacted by much more than the disease itself or the therapeutic intervention. And how does one really separate one from the other? So I think in designing registries and considering PROs, as I said, I think in the beginning, part of what I think a registry can really contribute to understanding natural history, or what the natural history can really help investigators and sponsors understand, are what is the potential benefit that patients are seeking with the illness, the rare disease, or the rare cancer that they might have, and then utilize that information to design, develop outcome assessments that can be standardized, validated, and used in a setting where the data collection and management, or the integrity of that is not going to be questioned. And I think going forward, that's what we would.
Great, thank you. Well, I'd like to thank all of our panelists for an engaging and very informative discussion. A number of these points will be taken up by our second panel. We heard about basic definitions of registries all the way through considerations of methodologies, and then the intents and uses of registries. Next, we're going to take a 5-minute break. And then after that, panel 2 will resume our discussion of rare cancer registries, specifically in the context of drug development. So we'll see all of you again in 5 minutes. Thank you.
So it's a great pleasure. My name is Zen, and my current role is the leading oncology at EMD Sprono. But today I have an honor to serve as a physician scientist to moderate this panel and work with my prestigious colleagues to take you through this topic David just mentioned, specifically what rare disease registry can help with medicine development. So before I start, I do want to, on behalf of my panelists, to take the opportunity to thank the organizers, Bill, Andy, Julia, to host this meeting during this challenging time and do this virtually. Many of us are frequent flyers here, been with the PDF FDA meeting over the past 9 meetings. I think most of us have been participating in each one of them because we love this meeting so much, giving the importance, the topic, the cutting edge topic, and most important thing, I think, like David said at the beginning during his keynote speech, it is unity. Because unless we have this open dialogue, this unity, we won't be able to advance medicine. Using my prestigious colleague, Mace Rosenberg always says, there's a speed of oncology because we want to develop a medicine for patients. So big thank you for that. Also, I do want to take a moment because this time we have this virtually. Many of the people are sitting in the audience, sitting in the audience as virtual participants. I do see Dr. Murphy there, and also the founding person, my colleague Mace Rosenberg. I do want to take the moment to thank them for starting this tradition and with Dr. Pasteur to enabling this very collaborative effort that really help us advance care for patients. So this specific topic of rare disease, very near dear to my heart as an oncologist by training, is I think that when we notice large indication, when we notice that the medicine development had to take a significant term in terms of acceleration, there is a population, there is a patient population that very difficult to reach and very difficult to conduct clinical trial with, and that's the rare cancer patients. And today's world, with the advancement of genetic, I would say capability, measuring the next generation sequencing, also there is a population called genetic or rare genetic alteration population. I still remember I used to read a book of N1 clinical trials. I mean, we're going to enter into an era that every patient's a rare cancer patient because the rarity of their background. So we have to treat at each patient's care with differences. And that's something I thought really, really important. Right now, we had this inflection point from both angles. One is, I think over the past 3 or 4 years, I'm going to pass on to the panelists. In terms of the progress we have made, I think it's the moment to celebrate a little bit of the success and the progress we made as a field. We see medicine approved based on the rare disease registry, real world experience, and also using that disease registry serve as a natural history, to Greg's point, to help the regulatory decision making. But also we wanted to look into forward to, I think my panelists, Michael Wong, challenge us, say, what about action? I think I really like this notion that also start by David, say, hope need to trigger action. I think this is something really important we will touch on at the end of our talk, and really also lead into the way of my great friend, Sean Cozen and Josh, to talk about a unique example of the fireside chat. So with that said, maybe we'll get started talking about speed oncology. What I'm going to do a little bit different from David Reed when we started is I'm going to let the panelists introduce themselves and then quickly touch on actually your recent success and experiences with the rare disease registry. There's no better person to start this with. Mike, I'm going to pass on the stage to you. Thank you.
Thank you very much for a kind introduction, and it's an honor and a pleasure and also a privilege to be on a panel with such esteemed colleagues and to be able to speak to you in general. I'm a professor at MD Anderson in the medical oncology department. I specialize in skin cancers and Merkel cell carcinoma in particular. I also serve a role as a medical executive within the MD Anderson Cancer Network, which comprises affiliate and partner hospitals throughout this country and the world. And in that capacity, I'm responsible for integration and special programs. And so I have a footing both in sort of a local thing where I touch patients. I had clinic just yesterday, and also more globally. I'm just going to say one word about this. And, I came to this in a way serendipitously. I was at a meeting that Dr. Brownlee from the NIH arranged for a workshop in Merkel cell carcinoma now almost 3.5 years ago. I stood in front of my colleagues and said that one of the major missing points is sort of an understanding of this rare disease, and that a registry and a database of sorts would be essential, and that I would not come back to the meeting if I didn't have one set up. So I'm very fortunate to be able to work with Dr. Miller, which you met in the previous panel, to really put this together. I'll speak more about this later in detail, but I'll pass the baton on to my colleagues for their introduction and remarks.
Greg, maybe we'll pass on that stage to you.
Again, it's a pleasure to be here. My name is Greg Freiberg. I am the lead of the development organization for oncology and bone at Amgen currently. I'm a medical oncologist by background. Had a short academic career before leaving for my industry role about 14 years ago. I've had the real pleasure in more recent days of working on some projects that leverage real-world evidence registry data, particularly in the ALL and the MRD, minimal residual disease positive subset, very rare cancers. And again, I think we're gonna get into these themes, but this concept of is that data there to supplement and complement, as Dr. Beaver I think pointed out very nicely, or can it be used for something else? And of course, the devil's in the details, and looking forward to having additional conversations in the panel.
Thank you, Greg.
Hi, good afternoon, everyone, and thank you so much for having me and my other FDA colleagues here to be part of this very important discussion. I'm a pediatric oncologist and the acting deputy director of the Division of Oncology 2 in the Office of Oncologic Diseases at the FDA. And within our division, we have the team that oversees the regulation of cancer drugs and biologics that are intended to treat pediatric patients with solid tumors, as well as patients with brain cancer and other rare solid tumor cancers. So this topic of rare disease registries, cancer registries, is really a very important one and near and dear to my heart. With respect to use of registries, I think others have talked about this already, particularly Julia and Greg Riemann. And we have had a very long history of using registries for use of trying to characterize safety, particularly in a post-market setting. Particularly when we're concerned about rare adverse reactions that may not have been adequately represented or even seen maybe in an initial database, which, it certainly can happen when you're looking at small data sets for patients with rare cancer. And so we've been using registries for that purpose for quite some time. And additionally, as already been mentioned, we have used registries many times to help inform the advice that we're giving to sponsors when they're developing drug development programs and clinical trials to help provide the data that will ultimately support a new drug application, to help us understand endpoints, provide advice on target effects, et cetera, in that context. And although these aren't examples of use of data from registries per se, we have had precedent for use of real-world evidence derived from data sources outside of clinical trials to provide additional evidence to support the approval of drugs to treat cancer. Notably, a couple just quick examples were in 2014, the approval of blinatumomab for the treatment of Philadelphia chromosome negative relapsed refractory pre-B-cell lymphoblastic leukemia. We used data in that approval not only from a single-arm clinical trial looking at complete response rate, but we also looked at data extracted from 2,000 patient records derived from data from the European Union and U.S. clinical study and treatment sites that gave us patient-level data, which provided very important clinical context for us to assess the meaningfulness of the CR rate observed in the clinical trial. So we use real-world evidence there in that context, and additionally, more recently, in the 2019 approval of Ibrance or palbociclib for the treatment of male patients with hormone receptor positive, HER2 negative, metastatic or advanced breast cancer. We use data from the Flatiron database to help document real-world response rates, and a small number of men with breast cancer, I think it was 20-some plus, But using that data, in addition to FAERS data to help provide some safety information, claims data were used, all within the context of what we already knew about palbociclib from randomized clinical trials in women, put together a data, and enabled the construction of a data package that ultimately led for, approval in this very rare, but important population of patients with breast cancer. So, I think all of those principles really extend to and speak to the importance and utility of registries, which I think provide even more potential in many ways to provide some structured data that we might need in the future, that we will need in the future.
That's really great. Yeah, and thank you, Marcia, for a nice, summary and also listed of the recent successes and how, from the agency was able to tap into this, a rare disease registry or real world data to enabling, I think, medicine development for some of the most rare cancer type we're seeing. Mike, you want to make a comment?
Yes, yeah. I think just to put a few things in perspective and a slingshot of what Dr. Reese said in a previous panel, and both Dr. Donahue and Dr. Freiberg spoke of specific patient population. In one, she mentions, failure of a chromosome. She mentioned a hormone receptor positivity or negativity. Dr. Freiberg mentioned minimal residual disease. Where I'm going with this is that we are in a world where it's going to get rarer and rarer, where we're going to personalize it down to the molecules. And if I learn one thing from Dr. Frankenbaum, there may be a Castleman disease, which is IL-6 negative, mTOR positive, VEGF receptor positive, ligand negative. And that's how we're going to start defining things. And if so, what's going to happen is that this is not just going to get better. This is going to actually get worse as we try to fractionate these drugs. And why do I even mention that? Because nowadays we sort of use medicines without biomarkers in the hope that it's going to work. And I built my career in immunotherapy, and we give immunotherapy to almost everyone who fits the clinical criteria. But we know that only less than half of them will have a long-term response. And if we only had a biomarker, I want to say not biomarker, biomarkers with an S. So as these things happen, we're going to need some mechanism to really flesh out who's what. I mean, this is sort of looking into the future, but I don't think it's going to be the distant future. So more than anything else, I think registries will become increasingly important, both post-marketing and in the initial drug development.
Love it, because that's the thing that we were excited because this is going to become a mainstream. I think it's no longer this unique one-off situation. We had to deal with say, open mind and also, some of the exceptions, but now it's going to become a mainstream. Greg, you want to make some comments? I think your recent experiences could also give a flavor to what the lessons learned and also some of the successes.
Sure. Thanks, Jen. Particularly with a disease like ALL, and particularly if you look at adults, it's a rare disease. Registries really have opened the opportunity to understand the disease in different ways. Of course, I kind of think of it in three bins. Any registry data, it'll have its challenges, and I think we'll get to that in answering a separate question. But they can be used to reflect on how the patients are doing in the past. You can think about which patients are being well served and which ones are absolutely not being well served by the therapies that are out there. And in the example I cite, being MRD positive, for example, is a very strong predictor. People who weren't okay, you needed to think about doing something. Similarly, when we're at the end of, say, we have experimental evidence in hand, you can think of this registry data, this external evidence, as being used to put that into context. I think we've discussed that already. There's a really, I think, clever way to use this data that's probably evolving, which is could you think about a way to use it as a prior evidence base to inform the actual what goes on in the middle, which is the testing of new therapies, to be prior information and in, say, a Bayesian approach, use it to either inform your control arm on a very basic level. We can use this data to, again, pick entry criteria. Think about who should come onto a randomized study if you can do it, and who should or should not be excluded. But really this idea of using the data in a more adaptive way is something that I know that several groups are thinking about. And again, it goes to show you, depending on what that data set is, where it comes from, how much confidence you have in it, you could think about using it in different ways. And Not to say that anyone is trying to make randomized studies go away. It's probably the greatest invention in the history of medicine is the randomized control trial. But there are just some settings where it will be impractical, if not impossible, because of numbers, where patients are, or patients aren't living long enough. Very sad practicality that you won't be able to get them to that therapy. So again, some thoughts off the top of my head, but I think ALL presented a nice case example where the first and the second, thinking about afterwards and beforehand, putting into context how you're going to design a study and then what it means were used. But the future will be using that data in again in the study and incorporating it into the statistical design.
Thank you, Greg. I'm going to write this down. The randomized control trial is still gold standard. maybe pass on that to Martha. Maybe let you give an agency's flavor in terms of how you see the world evolving. Martha, maybe I pass on to you and see how you view agency view where we are leveraging the real world data, either mainstream or how it can complement the mainstream.
Thanks. And I was half joking when I did this. And I appreciate, we do, of course, believe that the gold standard is a randomized control trial because it can control for sources of bias that we may not be aware of in particularly. It's really good at, when the sample sizes are big enough, it's really good at kind of, mitigating bias for factors we know about and, very importantly, ones that we may not know about that could impact patient prognosis. So, I think there are, many circumstances, many, where they're still very relevant and feasible to conduct. But, we do recognize at FDA that there are situations where randomized controlled trials are not possible. So either because the patient population is so small or because there's no good comparator arm and it wouldn't be ethical to randomize patients, for instance. And so, we've relied, in oncology, a lot on single arm trials, looking at sort of concrete evidence of tumor effectiveness stemming from response rate and durability of response, in particular when we're looking at rare cancer drug development. And so, the way I view use of registry data and other sources of real world evidence at this point in time is it's hard to make a general statement about it because it's a little bit really important. The context of use is important, and the particular situation, the particular disease is really important. And, and this is my own personal opinion, I don't yet see reliance solely on the use of registries at this point in time, as something that is likely to support a drug approval. But it's very easy to see, and we are already having multiple conversations and are overseeing drug development programs where real-world evidence from registry data and other sources is really providing a key and very important component to the data package that's contributing to what will hopefully be demonstration of the drug's effectiveness for rare cancers. So, I think in a particular situation where registry data and other real-world evidence data can be important is helping us interpret when there are circumstances where we can't rely on response rate, or we may need help interpreting what response rate really means to a patient. Having additional data that describes patient outcomes, both in patients who've received the drug or patients who have not received the drug, to help provide that context to us when we're interpreting the data that we have is crucial, and I think will be continuing to be leveraged more and more. There's been a lot of discussion and a lot of work thinking about using real-world evidence, as an external control, a synthetic control to help us interpret time to event endpoints in clinical trials. And I think there's a potential avenue, certainly for use of registry data for that purpose, although, it would have to be very, very, very carefully thought about and constructed, and, would require a lot of interaction and discussion on our part to make that, a critical component of a drug development program. So, I do see that there's going to be, as Greg, I think, mentioned in the last panel, I do think that there's going to be more and more focus on use of these types of data in the future. And I think particularly, now that we're looking at rarer and rarer cancers and rare subsets of rare cancers, which is driving, changes in our drug development paradigm. And I think thinking about use of registries in a global sense to capture data from as many patients as possible to give us, confidence in the results will certainly go a long way in helping that utility too.
That's really nicely said, Martha. You actually helped me in my job to open the next topic in terms of discussion about the challenges. So, and I know that Mike's very passionate about, sometimes, when you look at, to your point, Martha, the disease, why it's so rare, and make something impossible to do, find that many patients to do the randomized trial, and also sometimes, from timing, the cost, and everything, the efforts, they would not be, enabling a sense of urgency to accelerate. Because we know the cancer patient can't wait, and cancer is a lethal disease. So, and how to balance those two when the science advances very quickly, and we have a certain step to reach to a regulatory science to enabling understand the risk of benefit. And this has become an art, I would say. And then I look at Mike, probably you can give us your perspective of the challenges, particularly moving forward, because and also the dialogue between academic and the agency. So regarding how to overcome the challenge to a certain degree.
Sure. I when I was a much younger investigator and I had hair and a ponytail at the time, I see everybody laughs when I say that, including my kids. But at that time, we used to see, the FDA as an adversarial sort of agency, right? If only they let us do this, why can't I do that? But the reality is I've come to realize that this, it serves a very important role in sort of making sure the answer to two questions to me. Is it safe and is it true, right? And simply put, that's what it is. And we have to rise to that bar. And so I understand that. I also want to pick up a point that Greg built up, which is, these registries are not just sort of hypothesis testing, but they can be hypothesis generating in a sense, right? And our keynote speakers, sort of mentioned that in spades. And so, is there something down at the local CVS that we can see? So why do I say that? Because there are drugs that we know that overall survival in many of our patients is a function not just of what the test drug they got, but of the downstream care that they got subsequent to that. And as that improves, that can differentiate one arm over the other. So capturing that longitudinally, as Dr. Miller said in the previous panel, super important because now you start getting clues. And I, for one, believe that if you truly understand immunotherapy, sequencing matters. Where you go with your, is it a vaccine first? Is it going to be a checkpoint? Should it be a cytokine? Now we sort of give everything because that's all we got. But as we get more sophisticated, these things become increasingly important. And so the challenge is I have to be honest with you. I came to this because of the fact that I think it would be impossible to mount a reasonable phase 3 randomized control study in something like Merkel, which I treat, because of rare disease and also difficult getting patients and also the treatment benefit. Or you have a drug that's 10 times better, then of course it's a small trial. But I think the blockbusters and paradigm-changing drugs only come once in a while for ample reason. And because of that, everything else will be incremental. And you can imagine the number of patients necessary to make it happen. So know that will be really difficult to do. And lastly, I think there's a voice here that's not heard, but I think David Fangenbaum brought it out, which is the voice of the patient, right? They demand that we do something now. So how do we do this in the context of what we have in rare diseases? So the challenge is ours, right? And so one of the things Dr. Miller and I have been trying to put together, and particularly David, because I'm sitting on his shoulders and hope he's not cringing as I paraphrase him, is to really build sort of robust databases in collaboration with stakeholders like pharma, like the FDA. And really, it's like a limbo dance, right? How low can we go in the sort of quantity and perhaps quality of data that we get to satisfy regulatory requirements? And again, this is not adversarial, but we understand where you're coming from. How low can we go in the number of patients that we need, right? And are we able to build an adequate synthetic control arm? These are all challenges for us, right? I'll stop there. These are sort of the mainline things. There's of course, I have a basket of these things, but for me, these are the top line items, right? We have to solve the problem today, right? I had clinic yesterday, and we have patients coming in, and what trial they have for me now. So for me, I feel this pressure every single week as I see patients over and over again, and I'm just passing my anxiety to you. I don't have the answer, but here it is.
My job is simple. I'll pass that right to Greg. he's one I always can count on. Like how to overcome this, given that you have recently experienced this and brought in some success and also treatment options for ALL patients. Greg, how you look at these challenges, safety, efficacy, and also the quality and, inherent bias with the disease registry.
Let me pick up on that bias comment that you made. We often talk about how are we going to eliminate bias, but I like to use the sports analogy. You can never eliminate bias. You can only hope to contain it and understand it. And from that standpoint, really, when we're looking at these sources of data, of course, where you're getting the data from is absolutely essential. And we think about, well, what information is important? And I think, again, our keynote speaker very nicely laid out, loved his slide that had the 5 bins of the different types of registries. I stole it, and I'm going to use it in the future. It's just a very nice, I think, illustration that the choices you make will have consequences, whether it's on cost, on quality, or on numbers. But there are a few factors that I think about when you're looking at these registries that are probably universal. And one is that you have to make sure that you have the fields, that's the data information, the structured data, the fields that matter. And of course, what matters to you will depend upon what questions you're asking. But at the very least, since we're almost always asking time-based, how did these patients do over time? You need the basic prognostic factors for studies. Now, with ALL, for example, there's a big challenge, which is transplant is thrown in the middle, which often will transplant as many different things. And that will often scramble the patient's journey along the way and make it difficult to generalize. But 2 factors probably can help at least overcome some of that. One would be, and this was brought up very nicely by David Miller, reproducibility. Reproducibility, my statistician colleagues remind me, is again something, if you have multiple data sources that are telling you roughly the same thing, that's something that's much more powerful. And of course, size is our friend. The more data points you can have that fit the characteristics, the better. That's a luxury we don't always find, and we can't always take advantage of in the rare disease space. But those factors usually allow us at least to put this into context. I agree with Martha. It's really tough to talk about generalities here because each of these data sets and each patient has a different journey. But again, going very back to, I think, our keynote speaker and some of the introductions, the way you deploy this data, the way that you want to try to overcome challenges is absolutely depending on who you're studying and what you're trying to accomplish. But again, having that prospective view of it, laying out and seeing whether, again, reproducibility within different data sets can be harnessed is a key factor. Again, some generalizable ideas, but all politics are local when it comes to these registries.
I love it, Greg. I always take those nuggets from your talk is you can't eliminate bias, right? So maybe on my note first, given that the challenge with the bias in the package, the disease registry, and also the real world challenge sometimes with disease registry, that we won't be perfect. Any thoughts from your side can help the field advance, in overcoming this, bias and the challenges, so enabling a successful submission.
Sure, I can do that in 1 minute. No, Greg, I totally agree with you. I don't think our expectation is that bias is going to be eliminated. Absolutely, we understand. I don't think that you can guarantee that in most situations, even with clinical trials, actually. So the question then becomes how aware are we of these biases, and how are we going to address them, knowing that they exist, or quantify them in some way? So, I think for us, there definitely issues, that are important with respect to capturing as much information from patients in as consistent way as possible with as little missing data that is relevant to help us understand, how comparable are the patients, data that we're getting from a registry to the American public, for instance, and, are the data from the registry applicable to U.S. patients? Are they similar with respect to the underlying mechanism of the disease, the oncogenesis? Are they similar with respect to the types of treatment they received before getting, an intervention? After receiving the intervention, what, treatments happened afterwards? And could they be responsible for some of the effect we're seeing? And are those treatments comparable to what we would see, in U.S. patients? So, as with anything, having a clear picture about the patients themselves and their story, their clinical journey, helps us understand, if the data are applicable, and also if we're looking at this data, and comparing it to data from a clinical trial or other data, helps us gives us some sense as to whether we're comparing apples and apples to the extent that we possibly can. And having patient level data that's robust, that's capturing what we think is a generalizable patient population as possible, enables us to do analyses that will do propensity score matching to kind of help maybe account for any imbalances that we might see or any biases, or as we're sort of pressure testing the information. I think having that ability is really important for us to have in some cases. The principle of not fishing through a registry to get some information and then presenting it to us, rather actually having sort of this prospective development program where you have laid out the plan, this is the registry that we've chosen, this is why we think this registry is, or a combination of registries, are the best data sources, and this is how we're going to use this registry, in our, to show effectiveness, for example. Having all of that laid out and discussed ahead of time, also helps eliminate, some of the concerns that we have about getting at it. I think, not sure if it was Mike or Greg who said, get at what is truth. I mean, we all really want to know what is truth. Is this truly a drug effect or not? If this is a drug effect, how does this impact our patients? Is this making a benefit to our patients? So, having a strong understanding of the registry, having that transparency, I think, as Dave Miller was saying before, having all of that and having those discussions ahead of time help us, I think, best navigate, chart a course that I think we're as comfortable with as possible. I think when you have a big drug effect, that also helps overcome a lot of issues, right? So if we're talking about a marginal drug effect, then those issues become much bigger because then you have more uncertainty about, does this drug work or not? If on the other hand, you have a very large drug effect, then, sometimes some of those issues, melt away or become certainly less important.
That's really helpful. Thank you, Marcia. I think we also have some really interesting questions from the audience. So we do promise that to take on some of their questions. One question I think is really speaks to the heart and purpose of our job is helping patients. Here to talk about actually a perspective of patient enrollment and recruitment. And the question is, how do you envision patient registry could help for recruitment enrollment of patients for future trials? I think, as the industry side, Greg can speak this better than me, that we're encountering finding those rare patients, either, from an effort perspective and also screening perspective. It's not that easy to enabling a sizable trial. Marcia, to your point, to then enabling we can conduct a very thorough, I would say, hypothesis and experiments to find that signal. So any thoughts, Mike, since you're seeing patients on a daily basis, maybe you can give us your take on that.
I think in this age of social media, Twitter, Facebook, et cetera, I don't think that's as big a problem as you think it is. I'm incredibly surprised at how many patients show up in my clinic and tell me all about me because I heard about me from some other person on their blog or whatever. They are incredibly plugged in because their life is on the line. So they know which company is doing what and whatever. And I believe that they understand that going to a place which enables them to not just help themselves, but help every other person with this rare disease down the line will be very important. So you have a disease in which people can travel. Of course, there's some that you can't. And especially in a rare tumor, they'll go seek a second opinion. And they will travel to sites where they feel they're getting the best care. And they will travel to sites that feel that they have access not just to what you know, but what everyone in the field knows. So I truly believe in my heart and soul that yes, they're rare patients because the average is rare. But if you aggregate all these patients in this country and they are a force to reckon with, they'll come to the place that will make sense to them. So I think the challenge is to us to really construct the compelling trials to demonstrate to the patient. I'll give you an example. if you try to mount a placebo-controlled trial, my patients look me in the eye and shake their head and say, "You're kidding me, right? Right? I want them on the drug." I mean, for instance, so they kind of come with their own biases and their own, right? Greg, this is one of them, right? So with their own sort of wants and needs. And so a challenge is up to us to construct a compelling trial for these patients, to construct a trial that will answer critical questions and that gives them the best shot at a response. So I think we can do this. And you know, to have us all come together to present that to really the ultimate audience, which is our patients, is not that a challenge, but it's a task. It's a must-do task.
Brilliant, Mike. Thank you so much. And maybe that flipped to Greg. I mean, knowing that you know, this sort of, I would say, real world challenge. I mean, maybe to Mike's point, and now it's finally eased quite a bit in terms of enrolling those patients in the study. Will be interesting to see. I mean, there's an audience question regarding, you know, how to prepare this registration quality disease registry data sets. So because we know that quality is also important. So any thoughts on your side?
Just from the standpoint of using registries,
prepare a high quality data package.
Yeah,
for registration purposes.
You know, the registries can be helpful when you're designing your studies for registration. They can give you insights into who these patients are. And there are nitty-gritty factors like what prior therapies they had in reality and issues with con-meds, you know, really nitty-gritty things that we can help design our studies to be more inclusive rather than exclusive based on the registries. But the most important factor I think registries can get at is really there's a power, particularly when we have a molecular understanding of who these patients are. If there's a connectiveness to biobanks, to think about who are the right patients that we really want to be enrolling into our study. Where's the highest unmet need? And again, refining that population for whichever way it goes. Either they're being poorly served by what's out there, or potentially that they could be better served by this therapy. That would only have the opportunity to improve the effect size. And of course, nothing gets patients more excited to be on a study than seeing a good effect size of a drug. And so that's one, you know, I think one aspect that I feel quite passionate about, figuring out who the right people are to be on a study. We're not living in the age where histology is just defining the diseases anymore. And I think Mike and Martha have talked about this pretty nicely. And then finally, you know, getting patients onto studies from registries. I would say also that, you know, particularly the DNA sequencing companies that are out there, they're a form of registry in many ways. And they are trying to be used, whether it's as Mike points out, through a community, you know, creating a community for patients to give them information, or whether you're pushing information out to them through those channels. It's a way to get rare molecular subtypes on the studies. With regard to the making registrational data more, you know, higher quality, I just had one thought when Martha was talking I wanted to get back to, which was this idea of bias. How can you get rid of bias? Well, you know, we already said we can't get rid of bias. But if we have enough information and enough structured information, we can actually predict what missing data, if we're missing, how bad is it? And so, for example, in the ALL case that Martha was citing, in a related ALL case, you know, the data that we had allowed us to say something along the lines of, if 50% of the patients had an unmeasured variable, it would have to have an effect size bigger than any of the other known prognostic factors for it to change the result. And again, that gets back to Martha's point that big effect sizes are helpful as well. But I covered a lot of topics there, but wanted to at least loop back to that idea, which is we can quantify the impact of potentially.
That's so brilliant. And I know we have about 4 minutes left, and that means, you know, we're in the, you know, election season, that we have a poll on the side from the audience. Clearly, you guys done a fabulous job. Majority of the audience do see real-world evidence will become mainstream driver, more than 56%. I think the sample size is big enough to draw a conclusion there. And then the second thing is also they see this data can be used for synthetic control arm and the experimental arms for you know, consideration, you know, in the submission. So we won't leave this meeting without a call to action. maybe I'll go around the table and then again start with Mike, but then Greg and finish with Martha, see what your thoughts for the field to move forward. And probably in a minute time frame. Mike, go ahead.
Just very quickly, I feel like it's a presidential debate and I got 2 minutes. Just kidding. So you know, I'm filled with a sense of hope and optimism, and I want to pass that on to you because I see that things are moving. But in order for us to do that, we have to really come together as teams to develop compelling things that can help. I think other than us doing registries, the rest of the field has to come to us as well, including sort of new understandings of what sort of data is really needed and what sort of quality measures must be put in place. So we need a whole new set of standards. In addition, we need to engage with our mathematical colleagues. Are there statistical models that can help us understand bias? For instance, Martha said compare apple to apples. Well, maybe if we have registries that are spread so wide that it goes in the community and infiltrates every part of oncology, you can find the apple in a mass of oranges or something, right? So thank you. I thought you'd like that. I'm going to end there, but leave with a sense of optimism and hope because we are moving this chain. And if we ever have the next symposium on this topic, we'll come with data.
That's awesome. Thank you, Mike. Greg?
I would just add that, you know, as we think about this source of evidence, it's really important for the good of patients that of course, that we get as much as we can out of this. But the slippery slope is to take it too far and describe this data, you know, is not randomized information, and we have to be honest. And again, I think David Miller commented very closely, working with the experts in this field, whether that's statisticians or epidemiologists. There's a long history. I'm a physician, but that's not my expertise. We need to bring those people around the table to be able to fairly discuss what this data is and what it's not. Now, again, at one extreme, you have sort of the plural of anecdotes, which is not evidence. And at the other extreme, of course, we have information that can really help us not only design our studies, but interpret them on the back end. And of course, in scenarios where patients and time and dollars and physicians' time is just getting shorter and shorter, we have to make sure that we're asking the right questions in the right patients, and we're focusing on what matters. And that's where this data source can only help us.
Thank you, Greg. There's no better person than Martha to help us give a conclusion there a bit. Martha, you may be on mute.
I am. Sorry about that. But thank you for that. I get back to what Greg and, many people have said, before, is that, the purpose of registry is ultimately to improve the lives of patients. And so I think, one of the, things that, one of the calls to action needs to be, sort of continuing to carry the torch that PDS is carrying, and making sure that we have the opportunity to work together as a community to make sure that the registries are serving their intended purpose, and always keeping that in mind, bringing patients and advocacy groups to the table to kind of in partnership to make sure that these registries are resources that are used to the best of their ability to serve patients. So I think that to me is a really important action item. And I think a little bit hinging off of what Greg just alluded to, Dr. Pazder in the OC, one of his favorite things that he likes to say to us is, don't let perfect be the enemy of the good. And so, I think we have to keep in mind that, registries are not perfect. No matter what we do in the future, it's not likely, it's going to be messy to use to some degree data from registries and other real world evidence. And I think we have to accept that and work together to kind of, to make sure that the information is used in the best way possible. And I think it can be to expedite drug development, both for patients with rare cancers and other patients. So I'll stop there.
Thank you, Martha. That's a wonderful conclusion. And I want to take the opportunity to thank my wonderful panelists. You guys make the moderator job fun again. So thank you so much. Okay, so stay tuned. I think the next one is the fireside chat with my esteemed colleague, Sean Cozen and Josh Blinker. So we'll have a very short transition. So stay tuned. Thank you all.
It's a great pleasure for me to be having this fireside chat with Josh Billinker, who's the founding president and CEO of Loxo Oncology, which was recently acquired by Eli Lilly. Josh's unique blend of expertise is pretty special, and it includes deep regulatory experience as a former medical officer at the FDA, and a successful track record as a partner at Iceland Capital. And that has essentially placed him among the most effective leaders in the industry today. So Josh, it's great to be reconnecting with you, and thanks for joining the discussion today.
Thanks.
So, Josh, my first question is about Josh Billinker. Can you please tell us a little about his journey as a University of Pennsylvania trained oncologist who went from federal service to investing and then subsequently to launching one of the most innovative and efficient biotech companies in recent years?
Well, okay. First thing about Josh Billinker, never refers to himself in the third person. So here's a little bit about me. I was an aspiring drug developer in my last year of fellowship, and I came to the recognition that I was never going to be a great academic drug developer for reasons that I won't go on and on about. And I went to the FDA to work in order to understand the sort of final drive to the goal line of drug development. It felt like that was a black box that both PIs and other constituents just never really understood, and I thought, how interesting would it be to see how that really worked. I'm married to an attorney, and it's very common for attorneys to work at the SEC or the Department of Justice or elsewhere in the government to understand, how certainly regulated industries really work. And so I landed at FDA in the middle 2000s. At a time when the Office of Oncology hadn't yet been formed. It took form in the middle of the 2 years I was there. And it was a great place to get a lot of responsibility quickly, to learn on the ground, to understand how sponsors approach things. At the time, from a career trajectory standpoint, I had trouble seeing how, there was a many year arc, I thought at the time, between me and sort of some of the more senior leadership positions in the office structure then, and I think that's largely changed now, and I think it's a much more entrepreneurial culture than it was. Anyway, I started looking outside the walls for other opportunities, and I thought working as a venture investor would be a nice way to sort of have the fun of an energetic startup without committing to a single drug success. In other words, diversification across many companies by exposure. And then it was doing that for 7-8 years and living through the global financial crisis of 2008, among many other things, that I realized I was sort of more far removed from the drug development moment than I had expected or hoped, and I had a chance to start a company, and I did that from my desk in a New York City high-rise. And I'm sure we'll talk a lot about the drugs that ensued from there.
Yeah, that's great. So before we go into the specifics, the idea of precision medicine is something that people have been talking about for a long time, and typically it's defined as giving the right therapy to the right patient at the right time. So what does precision oncology mean to you in the context of a biomarker-driven approach to therapeutic development?
I think you said it in the last few words of the question, which is, to me, it's an incredibly diagnostic-driven conceptualization. It's a molecular diagnosis. It's I think in modern parlance, the use of tumor genomic profiling at scale to identify populations of patients who otherwise would likely not have ever been found, who then can be paired and matched with a unique single or hopefully cocktail of appropriate drugs.
Makes sense. So the way that we're diagnosing disease is evolving, and this came up during the last panel discussion. And it's very interesting when one thinks about that, how we've gone from classification of disease based on anatomy, where the tumor is, to histology, to now molecular profiling, where instead of the location of a lesion, we're looking at the underlying mechanism of disease. Obviously, Lux Oncology's larotrectinib spoke to those ethos and that strategy, and it was FDA's second tissue-agnostic approval. So do you think, Josh, that the next generation of opportunities in oncology need to think about pursuing a tissue-agnostic approach rather than taking everything to conventional ways of disease classification in oncology and the associated diagnostic modalities, and put more of our resources into understanding the underlying mechanism of disease and pairing our therapies to the mechanism of disease rather than histological classification or anatomic location of tumors?
I think it's just another tool in the drug development toolbox. And what I mean is that it's an incredibly sexy idea. It's an incredibly current idea, but it's not right certainly not right for every program. It's certainly not the easiest path to prosecute for most programs. In the case of the NTRK fusion story and the approval of Vitrakvi, it was really a development plan born out of necessity, and that necessity was the extreme rarity of the underlying biomarker. And it was there was really no credible way. We thought there were 20 disease types that all harbored at some small subset level an NTRK gene fusion that appeared to be activating an oncogenic in a context-independent way. And so, given the rarity, it was necessary to amalgamate, to bring together the 20 disease types and not get hung up on the sort of threshold statistics for each silo by histology, and instead use a biologic first principle and to say, well, I'm looking at the same genomic alteration. I seem to have point estimates which suggest that there's equal activity in a context-independent way. In other words, we don't have the BRAF melanoma, the BRAF colorectal exception, which everybody sort of points to. Mm-hmm. We don't seem to have that. And so how do we get critical mass of sample size? And the way to get critical mass of sample size to rule out a lower bound of clinically relevant efficacy was to bring together all these different histologies. And also, it was an age-agnostic approval. There was simultaneously approval in pediatric and adult patients. So that was another trick that was used to sort of gross up the sample size to, again, identify a clinically meaningful effect size. So it was born out of necessity, and it was born out of the diversity of the underlying tumor set, and it was born out of the extreme rarity in any given tumor type.
Sure, but it took a lot of insight, I'm sure, to recognize the necessity of it all. Because at the time, when you launched Lux Oncology, almost everyone else was focusing on immunotherapy and immune checkpoint inhibitors. So what was it that allowed you to see the path clearly and recognize the necessity, as you mentioned earlier?
Well, there were a couple of interesting things in the moment that struck me. So right around the time, within a couple of years prior to founding Loxo, in the sort of 2010 to 2013 period before Loxo existed, the ALK fusion story and the ROS1 fusion story in lung cancer had sort of unfolded, and that was a serendipitous finding of the sort of, as we all know now, what is the EGFR wild type non-smoker phenotype? Like, why do these look like EGFR tumors but they're not? Oh, well, there's these two other biomarkers at whatever we say now, 4% and 1% respectively, are explaining those tumors. And that nobody before that had thought that kind of prevalence was developable. Those were proof of concept programs in that they they took the bar lower and lower in terms of the underlying prevalence of the biomarker at hand. And I think between those three success stories in lung cancer, EGFR, ALK, and ROS, there was a rationale for like mass testing in lung cancer that began to unlock the long tail of less prevalent events. And then there was a Bob Doble's lab out of the University of Colorado. They published a study in Nature Genetics, a case series on the Foundation Medicine platform, where they identified, I believe, 3 NTRK gene fusion events in lung cancer patients who were pan-negative, pan-wild type for the canonical now EGFR, ALK, and ROS. So when I saw that, I was like, okay, here's yet another perhaps ROS1 type of development program. And so I thought when I committed to developing the TrkV and filed the IND, I thought it was going to become a lung cancer development program. And I thought it was going to be feasible because the lung cancer community had somehow already committed to the idea of testing genomically. And what I learned after about a year of enrolling no lung cancer patients was that, well, the underlying rarity in lung cancer was likely much higher than, or more extreme than the ROS1 rarity, which was already pretty low at 1%. So that just forced sort of a doubling down. And we got a bit lucky that the team at Foundation Medicine referred us a handful of patients. They sort of responded uniformly. The first patient was a sarcoma patient, had an amazing deep response after exhausting conventional therapies. So there was this flywheel of excitement that unfolded that to say, well, hey, maybe there's this pan marker that's rare in any given case, but sums to a clinically meaningful group of patients. So we sort of pivoted from a lung mindset to a pan-tumor mindset, and we were just hoping at that point that our colleagues at FDA would sort of see it the same way, and fortunately they did.
Sure. And so in terms of looking at what Foundation Medicine had in their data assets, did you have access to other registries to test out the hypothesis for a pan-tumor approach?
Access is a very strong word. No. I guess we had, I think the operational challenge that Loxo took on in its early days and did its best to excel at was just your question. How do we create the right set of relationships and alignments to find patients? And so I think there were 2 big sources of patients. One was the commercial central lab testing models of companies like Foundation and Caris and others. But the Foundation was ahead, sort of, certainly in its testing volumes. And then the sort of highly motivated tertiary care center sites like Memorial Sloan Kettering that were using tumor genomic profiling as a loss leader as part of their academic mission or by philanthropy support. And we're characterizing every patient who was being seen at the institution. And there aren't still today that many centers that embrace that approach. But identifying, so this is not a CRO-driven trial. This is a hand-to-hand combat. Because you think about the alignment you need. You need a molecular lab that's stood up the right tumor panel, that has the right probe sets to cover a gene fusion. In the case of NTRK, that was not a trivial problem. I mean, gene fusions, while I'd argue some of the most actionable events in oncology, are actually very difficult to find genomically unless you really try proactively. They take up a lot of sequencing bandwidth on DNA-based methods, and they're still not well tracked on blood-based methods, liquid biopsy methods. So you have to. So there's a chicken and egg problem. There's no reason for a platform to contain this difficult probe set if there's no drug, and there'll never be a drug if the panels don't include it. So there's this. You just sort of have to work with the serendipity that you're given to then generate exciting case series data that hopefully becomes exciting phase 1 data that then becomes more. And there's a proselytization that needs to happen. And Industry is not super well set up to pull this off, honestly.
Sure, it makes sense. In a way, if we're looking at precision oncology as tailoring treatments to patients who are most likely to benefit from the treatments, we're probably looking at many small subsets of patients, and our subsets are going to get smaller and smaller if we continue to make advances in the same direction. And you're absolutely right that the clinical trial enterprise and the way we conduct clinical trials, the enterprise, whether it's within one company or a CRO, isn't really set up to accommodate these types of studies. However, these types of studies are exactly the types of studies we need to bring tailored therapies to patients. Because if we think about, in the old days of double chemotherapy in non-small cell lung cancer, the number needed to treat was about 8. So we treated 8 patients for 1 patient to derive benefit. Right now, it's with small molecules and immune checkpoint inhibitors in the setting of biomarker-positive patients, it's about 2. That's amazing progress, but these are smaller subset of patients. Obviously, not every non-small cell lung cancer patient benefits from targeted therapies or would be eligible for targeted therapies. But number needed to treat of 2 is pretty impressive. It's amazing progress in the past couple of decades. But another way to think about that is that still half of the patients are not benefiting even from the targeted approach. And so there are smaller subsets within the subsets that may have a single genomic driver mutation. So do you think the answer to that is to look for multimodal biomarkers and complex biomarkers to tease out patients that still may not benefit maximally from a single biomarker-driven approach? Is that practical?
You're referring to the 1 out of 2 non-responders, or are you referring to the group that we have nothing for today?
Well, yeah. Well, let's say the patients that we typically say they have primary resistance. They just, they have the biomarker, but they just don't respond to the therapy. So there are probably other mechanisms, and the idea some people are advocating for a more complex phenotyping strategy. And in the world of immune checkpoint inhibitor, obviously we start with PD-L1, then microsite instability, and now there's tumor mutational burden. So it's already becoming, in a way, a complex phenotype. So is that the direction that we should pursue, in your opinion?
If you believe the literature that, say 10 to 20% of human cancers are these sort of simple, dumb ones of single gene driven, and the aspersion of response rates for those, whether, I think it's a function of the underlying drug, and, sort of average inhibitors that achieve IC50s get 40 or 50% ORRs, and the IC95 drugs get 75% ORRs, with another probably 10 to 20% missing resist cutoff, but clearly showing biologic dependence and probably deriving some benefit, even though we don't measure it very well in a strict regulatory sense. I don't think we have a big problem there, and I always find it funny that I have to answer for that question when I'm the guy with the 90% ORRs functionally. Where is the debate in the PD-1 panel? Why aren't we talking about the 10% who get durable disease control on PD-1 inhibitors when 100% of them are receiving the therapy? So I find it funny that I have to account for it. But I do care about the question of both primary and secondary resistance, and I think it's a translational commitment to serial biopsy that is ultimately the clue. And I think we've done a lot of work to look for on-target and off-target resistance, and on-target being, a binding mode problem, a mutation that affects relative potency. And then, of course, off-target is the, alternative pathway upregulation that would invoke a combination. So, interestingly, with sometimes a commercially available targeted therapy with a pathway that we know how to drug. And then there's probably another third or 40% where we'll never know. We don't know. And that group is frustrating, and that group, you know, in a high quality phase 1 center will have hopefully repeat serial biopsies that teach us the tumor biology at play. But I think we're doing a pretty good job there in the land of targeted therapy, and we could do even better if, again, these tumor genomic profiling was a more mainstream behavior. And it's still the rarefied technology of the, you know, the sort of, I don't know, leading 25 cancer centers. I mean, that's really where this is happening. I think the community will refer out liquid biopsy because it's kind of easy, but liquid biopsy misses a lot of the ultrastructural, like gene rearrangement stuff that is highly actionable. And so, you know, I somehow we have to cross that chasm, and I have this sort of pragmatic, fatalistic view that the only way it really happens isn't through conferences, it isn't through medical education, it's through better reimbursement. Labs need to be motivated and incentivized to run these tests more often and faster.
Makes sense. So speaking of reimbursement and a closely related concept, drug pricing. So how do you think those debates are going to evolve in the next 5 years? There's clearly a lot of focus on drug pricing. And what are some strategies that you think we can employ in order to you know, have a open and transparent discussion about drug pricing, tying it to perhaps outcomes? You know, folks typically pricing in for many biopharma companies is typically a peri-approval process kind of thought, you know, when you're close to getting the approval and, yes, in some cases even, you know, post-approval. However, you know, there are folks that now are talking about pre-contracting negotiations with third parties doing development. So what are your thoughts about that, and how can we get a better handle on drug pricing?
Well, I think I'm glad you didn't use the term financial toxicity because I think that's a deliberately sloppy term that conflates two separate problems. One problem is the out-of-pocket exposure of a sick patient to a non-discretionary choice, which is to save their own life by taking a drug. So why should they be penalized with inordinate out-of-pocket exposure to that drug, no matter what it's priced? And there's really no price for a drug that I think is fair or equitable to more than half the population that has virtually no savings or long-term financial security. So, and I think the second issue is the one you're asking about, which is drug pricing. And so, are we willing, as a clinical community, to talk about how much a year of life is worth? We used to be. We did it in the 80s when, you know, hemodialysis needed to be covered by Medicare, no matter, even under the age of 65. We used to talk about 55,000. You know, in the British system, NICE talks about it in the quality adjusted life year framing. And so we don't really, you know, contractors who were sent to Iraq to work in a war zone were given a hazard pay bonus of $250K, implying, you know, a year of life to them was worth $250K. And so how do we, like, this is a collective conversation we all need to have. And I don't see any of us really jump into that pool. I think we're all doing this head fake of like, well, it's all a reference-based conversation, like drug importation, European pricing, you know, well, like somebody's setting a reference, and can we have an evolved conversation about what that should be? And I've said this before in other settings, but I think if you tell the community this is what we're willing to pay, I mean, drug development is a 5 to 10 year lead time endeavor. So, and it takes, you know, Vitrakvi, which addressed the very rare population, we spent, I believe, over $400 million getting it approved on a database of 55 patients. And we did it on a team of less than 100 people, largely. And I think we did it about as lean and as efficiently as possible, and it was still a $400 million process. We weren't spared any expense in terms of CMC, standards, in terms of data quality standards, in terms of global regulatory and quality scrutiny. And so the fixed costs of the equation aren't different for the small drug and the big drug. And I think the investor community looks at the unknown of a 5 to 10 years out problem, and it says, well, you're asking me to put this money at risk with a go to zero probability because a lot of drugs fail. Like, what is the risk reward of that bet you're asking me to make? And I think it's not good enough to do it in the peri-approval setting because the money's already sunk then. The capital's always been put at risk, and that's not really fair. And the investor community will get wise to that and just not play the game. And I think that's already happened, by the way, in medical devices and diagnostics, where funding really isn't happening nearly to the same degree because there's no price transparency at the time of investment risk. And so you asked me about the political debate we should have. Hopefully it's more nuanced, less coarse, and more compassionate than the one we have collectively about every other issue in our society. This one's really important. Like, we've got to get this right. There's no other modality in the healthcare system like drugs that can bend the curve from a disease outcome standpoint. And I mean, there's really nothing out there like it. You can't say that about durable medical equipment or end-of-life care or, facilities and things like that. So, sorry to ramble on about your great.
Sure. Now this is
you're passion for it. Yeah.
Sure. I mean, you're making great points, and I recently read the great American Drug Deal by Peter Kolchinsky, who's a founding partner at RA Capital. He made a great point about how much we pay as a society for branded drugs. And he made the argument that sometimes we mistake paying mortgage for paying rent. Rent is an expense. However, paying mortgage, you build equity. It's an investment. And paying for branded drugs is more like paying mortgage because you're building equity, and these drugs ultimately go generic, and the price drops. So I think you're absolutely right that in order for us to advance and to be able to have those discussions, we need to collectively have more open and transparent discussions, and patients obviously have to be involved in these discussions because ultimately we want to reduce the burden on the patient, and out-of-pocket costs don't always correlate with the price of the therapy specifically. So no, you weren't rambling, and these are very important topics that maybe there'll be another opportunity in the future for us to dissect out those themes because they are critical in terms of how we think about drug development in the next 5 to 10 years. So we're almost out of time. I have one last question, Josh, which has to do with what you mentioned earlier in terms of the challenges in patient recruitment and the types of trials that we need to set up for patients with rare cancers. Obviously, there are modalities and the things that we may be able to do with rare cancer registries, constructing external controls, but the idea of how we conduct clinical trials. What are your thoughts about decentralization of clinical trials as a way of reaching patients who do have rare diseases and rare cancers? And because when we think about it, even in non-rare diseases and in oncology, only about 3% of adult oncology patients have access to conventional clinical trials. So do you think decentralizing clinical trials and leveraging the technologies that we have available to us today for remote monitoring, remote data collection, is a practical way of extending the reach of clinical investigations to greater groups of patients, including patients with rare cancers?
I don't think it's practical yet, for two reasons. The patients who live far away from the tertiary center are also likely ones who don't have access to genomic profiling. So they're sort of both far away geographically and far away scientifically. and both are a problem. I think the second is, I mean, I'm being deliberately provocative a little bit because I know that there's a lot of FDAers involved in this conference, but I haven't seen FDA really get comfortable with the more remote elements of data collection. Is FDA going to be okay with the lack of central radiology review? Is the FDA going to be okay with fewer study visits, going down from, say, weekly labs to every 6 to 8 week labs? Is that going to be okay? Are they going to be okay with sort of maybe even less robust data quality collection because we're dealing with a less sophisticated investigator community that's not a professional trialist community? Like, I just don't, if the end goal is drug approval, which hopefully it is if you're running a study, I think it's hard to really get comfortable with the trade-offs from a data quality perspective. I think the FDA is going to have to teach us how to do that and convince us that they're really ready for us to be ready.
Sure. Josh, we're out of time. Thank you so much for joining the discussion today. I think we could talk for another couple of hours, but next time.
Thanks for having me. It's a lot of fun.
Thank you, Josh. Coming up next is Dr. Andy Crichton, CEO of the CEO Roundtable on Cancer and Project DataSphere.
I want to thank you for attending Symposium IX, focused on rare tumor registries. I also want to convey our deep appreciation for the time and knowledge shared by Dr. Feigenbaum, Dr. Belinker, and the panelists today. Thank you to the PDS staff for all the work in preparation and handling behind-the-scenes work related to coordinating today's webinar. Please complete the poll that is on the screen, and we will be posting links to the recordings in our website, projectdatasphere.org, and we will send an email to everyone who is registered. I want to share Mike Wong's optimism and look forward to him coming back to the symposium when we're on this topic again with data. Precision medicine will increase the number of rare cancers, as common cancers will have rare components and need to be tracked. We see rare tumor registries becoming an important component of our external control arm stream of work. We should remember the goal is not only to identify new therapeutics, but to look for opportunities to repurpose existing therapeutics. Curated data for registries that is important to make it usable and bring consistency among institutions. Curation of data is costly and resource intensive, especially to capture unstructured data. We see the need for technology to efficiently pool this structured and unstructured data to become a game changer that needs to be fully investigated. Finally, we need to continue working together as a community, keeping the patient in mind. So again, thank you for joining us today and look forward to your comments. Have a good day.